There is a creature alive today that carries the blood of the most terrifying animals that ever walked this earth, and you have almost certainly eaten one. It lives in barns and backyards. It clucks and pecks at the ground. It lays the eggs you crack open on quiet mornings. And somewhere buried deep inside its genome, folded into the invisible architecture of its cells, is a story so ancient and so strange that it will remake the way you see the world around you. How does a monster become a bird? How does 66 million years of silence become a feather, a beak, a warm body nestled in straw? What did the last common ancestor of the chicken and the Tyrannosaurus feel when it opened its eyes for the very first time? These are not small questions. These are the kinds of questions that science has only recently begun to answer, and the answers are more astonishing than anything imagination alone could manufacture. Tonight, as the darkness settles around you and your breath grows slow and even, we are going to follow that story from its very beginning, through fire and extinction and impossible survival, all the way to the animal that shares your breakfast table. Let us begin. There was a time when the earth did not know silence. For more than 165 million years, the dominant land animals on this planet were the dinosaurs, a group so varied and so successful that they filled every ecological role available to them, from the smallest insect-chasing runners to the largest animals that ever stood on solid ground. The word dinosaur comes from the Greek words meaning terrible lizard, but this name is misleading in almost every way that a name can be misleading, because dinosaurs were not lizards, and many of them were not terrible at all. They were, in the truest sense of the word, animals with blood that moved through their veins, with hunger and instinct and the same basic cellular machinery that drives every living thing on Earth today. The non-avian dinosaurs, meaning all the dinosaurs that were not birds or the direct ancestors of birds, represent one of the most extraordinary chapters in the history of life on this planet. Their bones have been found on every continent, including Antarctica, and their fossils speak of a world so different from our own that it seems impossible it ever existed. And yet it did exist, and it lasted far longer than our own species has any right to expect to last. The sheer duration of their reign is something the human mind struggles to hold. The earliest dinosaurs appeared approximately 230 million years ago, during a geological period known as the Triassic, and they did not begin as the giants that most people imagine when they close their eyes and think of prehistoric life. The first dinosaurs were small, bipedal animals, often no larger than a modern turkey, that scurried through a world already crowded with other reptiles and early mammal-like creatures. What gave the dinosaurs their eventual dominance was not brute size, but a set of anatomical advantages. Chief among them, an upright posture made possible by a distinctive hip structure that allowed their legs to be positioned directly beneath their bodies, rather than sprawling out to the sides, the way a lizard's legs do. This upright stance made them more efficient movers, more capable of sustained locomotion, and more adaptable to a wide range of environments. Within a few million years of their first appearance, following a mass extinction event at the end of the Triassic period that eliminated many of their competitors, the dinosaurs began to radiate outward into every available ecological niche, with a speed and thoroughness that left the world permanently changed. What had begun as a modest experiment in reptilian anatomy became, in geological terms almost overnight, the ruling class of an entire planet. The dinosaurs are formally divided into two major groups based on the shape of their hip bones, and understanding this division is essential to understanding how birds eventually emerged from their ranks. The two groups are called the Saurischia and the Ornithischia, which translate roughly to lizard-hipped and bird-hipped, and the naming contains one of the great ironies of paleontology. You might expect that birds descended from the bird-hipped dinosaurs, and yet the opposite is true, because birds are now understood to have descended from the lizard-hipped group, specifically from a subgroup within the Saurischia called the theropods. The theropods were predominantly bipedal, meaning they walked on two legs, and they included some of the most famous animals in the fossil record, among them Velociraptor, Allosaurus, and Tyrannosaurus rex. The bird-hipped ornithischians, despite their confusingly named anatomy, included animals like Triceratops and Stegosaurus, and left no living descendants at all. This taxonomic puzzle, the fact that birds came from the lizard-hipped group rather than the bird-hipped one, was one of the clues that kept scientists from recognizing the true nature of the bird-dinosaur connection for so long. Nature does not always arrange itself according to what seems logical from the outside looking in. The sauropods were the largest animals ever to walk on land, and they belonged to a branch of the Saurischia that was separate from the theropods. Yet they share a common ancestor with the animals that would one day become birds, and their existence illuminates something profound about the plasticity of the dinosaurian body plan. Animals like Brachiosaurus and Argentinosaurus reached lengths of 30 meters and weights of perhaps 70 tons, achieved through a skeletal architecture that was both massive and surprisingly hollow. Paleontologists have long known that sauropod bones contained air spaces, a feature called pneumatization, and this was once thought to be simply a weight-saving adaptation that allowed such enormous animals to move at all. But the discovery that modern birds also have extensively pneumatized skeletons, with hollow bones connected to a system of air sacs that extends throughout the body, revealed that this feature was not invented separately in each group. It was inherited from a common ancestor, passed down through tens of millions of years of evolutionary change. And the air that flows through a chicken's bones today is a structural echo of the air that once moved through the bones of the largest animals that ever existed. The breath of a giant lives inside the smallest of its descendants. Tyrannosaurus rex is perhaps the most famous animal that has ever lived, extinct or otherwise, and the science surrounding its biology has undergone a revolution in the past 3 decades that would have seemed impossible to earlier generations of paleontologists. T-Rex lived approximately 66 to 68 million years ago in what is now Western North America, and it grew to lengths of 12 meters and weights of roughly 9 tons, making it one of the largest predators in the history of life on Earth. For most of the 20th century, T-Rex was depicted as a cold-blooded reptile, slow-moving and sluggish, more like a giant, scaly lizard than anything we might recognize as an active hunter. But evidence accumulated slowly and then all at once, through the study of bone microstructure, isotope chemistry, and eventually molecular biology, that T-Rex was almost certainly warm-blooded, that it grew at a rate comparable to large modern mammals, and that it may have been partially covered in feathers or feather-like structures, particularly during its juvenile stages. The monster of popular imagination and the actual animal described by modern science are two quite different creatures, and the real one is somehow even more extraordinary than the fantasy. To know an animal accurately is always more wondrous than to know it through invention. The growth rate of Tyrannosaurus rex was one of the first pieces of evidence that forced scientists to reconsider everything they thought they knew about dinosaur physiology. And it was revealed not through any spectacular fossil discovery, but through the patient examination of the internal structure of bone. When paleontologists cut cross-sections through T-Rex bones and examined them under a microscope, they found a pattern of growth rings not unlike those found in tree trunks, which allowed them to determine the age of individual animals at the time of death. What those rings revealed was shocking, because T-Rex did not grow slowly and steadily the way a cold-blooded reptile grows, adding a small increment of bone tissue each year across a long lifespan. Instead, T-Rex grew explosively during a period of adolescence, adding as much as 2 kilograms of body mass per day during peak growth years, a rate that is simply not possible in a cold-blooded animal with a slow metabolic engine. This kind of rapid growth requires the sustained production of enormous amounts of energy, which requires a warm body, which requires a metabolic system fundamentally more like that of a bird or a mammal than like that of a modern lizard or crocodile. The bones told a story the fossils alone could never have told, and that story turned a monster into something almost familiar. Feathers are among the most complex structures produced by any animal on Earth, and the discovery that they were not invented by birds but inherited from their dinosaurian ancestors is one of the most transformative scientific revelations of the past century. The first definitive evidence that non-avian dinosaurs could bear feather-like structures came from Chinese fossil sites in the 1990s and early 2000s, particularly from the Yixian Formation in Liaoning Province, where exceptionally fine-grained sedimentary rock preserved soft tissues that would normally disappear entirely during fossilization. Animals like Sinosauropteryx, Microraptor, and Anchiornis were found covered in structures ranging from simple filaments to complex branching forms indistinguishable from the feathers of modern birds. These fossils did not merely suggest a connection between dinosaurs and birds. They demonstrated it with a clarity that ended any serious scientific debate. Feathers did not begin as flight structures. They began, current evidence suggests, as insulators, as display structures, possibly as aids in brooding eggs, and they were present in the ancestors of birds tens of millions of years before flight itself evolved. The wing of a bird is not an invention, but a transformation of something that was already there, waiting to become something else entirely. The brain of a dinosaur is something that can be studied in surprising detail without ever having found a single neuron, because the brain cavity leaves its impression in the surrounding bone, and from casts of that cavity, known as endocasts, scientists can reconstruct the general shape and relative size of dinosaur brains with considerable accuracy. What these endocasts reveal is a wide spectrum of neural complexity, ranging from the relatively small and simple brains of large sauropods to the proportionally large and elaborately structured brains of small theropods. The small, bird-like theropods, particularly those belonging to the group called the Maniraptorans, which includes the ancestors of birds, had brains that were notably enlarged relative to body size, with expanded regions associated in modern animals with vision, balance, and coordination. The cerebellum of these animals, the brain region responsible for coordinating movement, was enlarged in a way that closely resembles the cerebellum of modern birds, and the optic lobes, responsible for processing visual information, were similarly prominent. These animals were not operating on instinct alone. They were navigating a complex world with a complex brain, and the descendants of those brains eventually became the minds of the birds that sing outside your window in the morning, the same windows behind which you are now resting. The reproductive biology of non-avian dinosaurs has been a subject of intense scientific interest and considerable surprise, particularly in recent years, as the fossil record of eggs, nests, and embryos has grown rich enough to allow meaningful analysis. Dinosaurs laid eggs, as their reptilian ancestors did before them, and as their bird descendants do today, and the eggs of different dinosaur species varied enormously in size, shape, and shell composition. But it is not the eggs themselves that have most surprised paleontologists. It is the evidence of nesting behavior, because the fossil record now contains numerous examples of adult dinosaurs preserved in postures that can only be interpreted as brooding, sitting on or beside nests of eggs in the same posture used by modern birds incubating a clutch. Oviraptor, a small theropod whose name means egg thief, and which was originally named based on a misunderstanding, was in fact found sitting atop a nest of its own eggs, arms spread over them, protecting them in death as it had in life. This brooding posture requires not just the behavioral instinct to remain with eggs, but a body temperature warm enough to actually incubate them effectively, which adds another layer of physiological evidence to the growing case that many theropods were warm-blooded and bird-like in their biology. Every nest is a letter written to the future, and some of those letters were written in stone. The arms of theropod dinosaurs are one of the most revealing features in the entire fossil record of the dinosaur-to-bird transition, because they preserve, in bones that are hundreds of millions of years old, the anatomical precursors of the modern bird wing in extraordinary detail. Among the most important features is the wrist structure, specifically a bone called the semilunate carpal, which in many theropods allowed a distinctive folding motion of the hand that is directly analogous to the wing folding motion seen in modern birds. When a bird folds its wing, it does so by rotating the wrist in a specific way that brings the long primary feathers close to the body, and the same rotating wrist mechanism was present in Velociraptor, in Deinonychus, and in many other non-avian theropods, long before any of them were flying. These animals were not using this wrist motion to fly. They were using it for something else entirely, perhaps to pin prey, perhaps to climb, perhaps for display, and the movement that eventually became essential for flight was borrowed from a functional context that had nothing to do with the sky. Evolution is endlessly opportunistic in this way, taking structures evolved for one purpose and finding in them the raw material for something completely different. The wing began as a hand. The respiratory system of dinosaurs, reconstructed through careful comparison with modern birds and crocodilians, the two living groups most closely related to non-avian dinosaurs, was almost certainly more efficient than the tidal breathing system used by most terrestrial vertebrates, including humans. Modern birds breathe using a system of air sacs that allows air to flow unidirectionally through the lungs, rather than in and out in the tidal pattern of mammalian breathing. And this means that fresh, oxygenated air is always present in the gas exchange surfaces of the lung, never mixed with the stale, depleted air that mammals must push back out through the same passages it entered through. This system provides exceptional oxygen extraction efficiency, which is why birds can fly at altitudes where mammals would quickly lose consciousness. Comparative anatomy and bone pneumatization, patterns in theropod dinosaurs, strongly suggest that many of them, including the large-bodied predatory theropods, possessed a similar air sac system, which would have given them respiratory capabilities far beyond what their body size alone might suggest. T. rex, stalking through a Cretaceous forest, may have breathed in a way more similar to a modern sparrow than to a modern crocodile. The dinosaur and the bird share not just their bones, but the very rhythm of their breath. The skin of non-avian dinosaurs has been a matter of fierce scientific debate and progressive revision, as new fossil evidence has forced one reassessment after another of what these animals actually looked like in life. For most of the 20th century, dinosaurs were depicted with smooth, scaly reptilian skin, and this depiction was not entirely wrong, because large-bodied dinosaurs like Tyrannosaurus rex did possess scales on many parts of their bodies, as preserved skin impressions have confirmed. But the picture is considerably more complicated than a simple coat of scales, because the same evidence that revealed feathers in small theropods also revealed that feather-like structures were present on many lineages, and that some large-bodied tyrannosaurids may have had feathered juveniles that became predominantly scaly adults, a pattern somewhat reminiscent of the way some baby birds lose their downy covering as they mature. The scales of dinosaurs and the scales of modern birds are not the same structure, however, because bird scales, found on the legs and feet, are now understood to be feathers that have been developmentally suppressed, reduced back to their ancestral form by genetic mechanisms that turn off the complex branching program of feather development. The foot of a chicken, scaly and reptilian-looking, is not evidence of a simpler time. It is evidence of a complex developmental system capable of playing its own genetic history in reverse. The sensory world of theropod dinosaurs was almost certainly dominated by vision, and the evidence for this comes from multiple independent lines of inquiry that converge on the same conclusion with remarkable consistency. Eye socket size relative to skull size, preserved scleral ring bones that allow estimation of eye diameter, and brain endocast geometry all point to theropods as highly visual animals with large eyes and substantial neural investment in visual processing. Comparative studies of the eyes of modern birds and crocodilians, both of which have a much richer color vision than most mammals, including humans, suggest that the common ancestor of all these groups likely had tetrachromatic vision, the ability to see 4 types of color, including ultraviolet wavelengths invisible to the human eye. If theropod dinosaurs inherited this tetrachromatic system, and there is no particular reason to think they lost it, then Velociraptor and its relatives were seeing a world painted in colors that no human has ever directly perceived, a world where the feathers used for display blazed with ultraviolet signals invisible to our kind of eyes. The Cretaceous landscape that we imagine in grays and greens may have been, to the eyes of its inhabitants, a world of impossible vividness and color. We see only a faint shadow of what they saw. Social behavior among non-avian dinosaurs is one of the most contentious and fascinating areas of current paleontological research, because it touches on questions about the inner lives of these animals that the fossil record can answer only obliquely, through the interpretation of trackways, bone beds, and nesting sites rather than through direct observation. The evidence that some dinosaurs were social animals is actually quite compelling, particularly for certain herbivorous groups, where massive bone beds containing hundreds or even thousands of individuals of the same species suggest that these animals lived and died in large herds. But social behavior in theropods is less clearly documented and more debated, and the question of whether predatory dinosaurs like T-Rex were solitary hunters or pack animals remains genuinely open. What is clear is that some small theropods, particularly those most closely related to birds, show evidence of extended parental care, which requires at minimum the maintenance of a social bond between a parent and its offspring across a period of time. And this kind of extended juvenile dependency is a hallmark of the most cognitively complex animals alive today. The dinosaur that stayed with its young was investing in the future in a way that simpler animals do not, and that investment echoes forward through every chick that huddles beneath its mother's wing. The immune system of non-avian dinosaurs left almost no direct trace in the fossil record, yet paleontologists have found something unexpected and poignant in the bones of individual animals that speaks to the biological challenges these creatures faced during their lives. Bones from multiple theropod species, including several T-Rex specimens, show evidence of healed fractures, infections, tumors, and parasitic infestations, all preserved in the internal microstructure of the bone tissue. One particularly famous T-Rex specimen shows evidence of a bone infection so severe that it likely contributed to the animal's death, caused by a pathogen related to modern trichomonas, the same parasite that causes disease in modern pigeons and other birds. This is not a trivial observation. It is a direct biological link between the immune challenges faced by T-Rex and the immune challenges faced by living birds, a shared susceptibility that hints at shared biological machinery in the systems that protect the body from invasion. The body of a dinosaur was not an armored fortress immune to microscopic attack. It was a warm, living system navigating the same invisible biological battlefield that all warm-blooded animals must navigate. The same battlefield that a chicken navigates today when it shakes off a dust bath and tucks its head beneath its wing. The metabolism of non-avian dinosaurs is perhaps the single most debated topic in all of paleontology, and the reason the debate has been so heated for so long is that the answer has profound implications for everything else we think we know about these animals. Their behavior, their ecology, their growth, and their ultimate vulnerability to extinction. Cold-blooded animals, technically called ectotherms, rely on external heat sources to regulate their body temperature and have low metabolic rates that allow them to survive on very little food, but also limit their activity levels and growth rates. Warm-blooded animals, technically called endotherms, generate their own body heat through metabolic activity and have high metabolic rates that require substantially more food, but allow sustained high activity and rapid growth. For most of the 20th century, dinosaurs were assumed to be ectotherms, essentially giant reptiles, but the evidence accumulated over the past several decades has overwhelmingly shifted scientific consensus toward the view that at least the theropods, and possibly most dinosaurs, were mesotherms or full endotherms, generating significant body heat internally and maintaining metabolic rates well above those of modern reptiles. The evidence comes from bone microstructure, from isotope chemistry, from growth rates, and ultimately from the fact that birds, which are unambiguously endothermic, are living dinosaurs. The warmth inside a bird is not a new invention. It is an ancient inheritance. the geographic distribution of non-avian dinosaurs across the supercontinent known as Pangaea, and its subsequent fragments, tells a story of both connection and isolation that shaped the evolution of the group across its entire history. When dinosaurs first evolved in the Triassic, the world's landmasses were largely united into a single supercontinent, and this allowed the earliest dinosaur lineages to spread across the entire planet with relatively few barriers. As Pangaea began to break apart during the Jurassic and Cretaceous periods, the separating landmasses carried their dinosaur faunas with them, and geographic isolation promoted the evolution of distinct dinosaur communities on different continents. North America and Asia, however, remained periodically connected by land bridges, and this allowed a sustained exchange of dinosaur lineages between the two landmasses across much of the Cretaceous period. Tyrannosaurus rex and its close relatives, for example, appear to have had Asian origins, crossing into North America during one of these periods of connection, and diversifying there into the apex predators of the late Cretaceous. The continents that now seem so fixed and permanent were, on the timescale of dinosaur evolution, moving and connecting and separating like slow-motion puzzle pieces, and every shuffling of those pieces reshuffled the biological communities living on them. The world that gave birth to the ancestors of the chicken was a very different world from the one the chicken knows. The extinction of the non-avian dinosaurs 66 million years ago was not the simple, sudden event that popular culture has made it seem, and understanding its actual complexity is essential to understanding why birds, alone among all the large animals of the Cretaceous, survived to inherit the Earth. The extinction event was triggered by the impact of an asteroid approximately 10 to 15 kilometers in diameter that struck what is now the Yucatan Peninsula of Mexico with a force equivalent to billions of nuclear weapons, releasing energy sufficient to ignite wildfires across entire continents, inject massive quantities of debris into the atmosphere to block sunlight for months or years, and trigger a cascade of chemical changes in the oceans and atmosphere that unraveled the food webs on which all large animals depended. But the impact did not kill every dinosaur instantly and simultaneously. It killed by disrupting the systems that sustained them, and the disruption played out over years and decades following the initial event. Different species in different places, with different ecologies and different behavioral repertoires, survived for different lengths of time, and the survivors, the birds, were those that happened to possess the combination of traits, small body size, dietary flexibility, and rapid reproduction, that allowed them to navigate a world in which the rules had suddenly and catastrophically changed. Survival is not always about being the strongest. Sometimes it is about being the most adaptable when the world no longer rewards strength. The fossil record of the latest Cretaceous, the final few million years before the extinction event, is rich enough in some regions to allow paleontologists to assess the diversity and abundance of dinosaur communities immediately before the catastrophe struck, and what they find there is both illuminating and sobering. In the western interior of North America, one of the best sampled regions for late Cretaceous terrestrial life, the dinosaur communities appear to have been diverse and thriving right up to the very end, with no clear evidence of a long, gradual decline that preceded the extinction. The Chicxulub impact, as the asteroid strike is formally known, hit a world that was biologically robust and shows few signs of having been weakened in advance by any preceding environmental deterioration. This matters for the bird story because it means the bird's ancestors did not survive because the dinosaurian world was already collapsing, and they simply outlasted it. They survived through something far more active and contingent than that. They survived because they were, in some combination of ways that we are still working to fully understand, the right kind of animal for the wrong kind of moment. And that survival, against every expectation and every probability, is what eventually put a chicken in your kitchen and a feather in the morning air. Let the weight of those ancient years settle gently into your body now, like sediment drifting down through still water to the quiet floor below. The world of the dinosaurs was long and loud and full of motion, and you have carried it with you through every word. Your breath is slow now, and the images of those ancient animals are growing softer, less sharp, like the edges of a dream that has not yet decided what it wants to become. The Cretaceous sun is setting somewhere behind your eyelids, and the forest is going quiet, and somewhere in that quiet a small feathered creature is tucking its head beneath a borrowed wing and beginning, without knowing it, the longest journey in the history of life on Earth. You do not need to hold any of this. Let it rest. Let it breathe. Let the story continue to unfold on its own, slowly, as you drift a little deeper into the comfortable dark. The asteroid that ended the age of dinosaurs was not a unique object in the solar system, but rather one of countless rocky bodies left over from the formation of the planets. A fragment of the early solar system that had been orbiting the sun for more than 4 billion years, before it finally intersected the orbit of Earth on a morning 66 million years ago. It arrived without warning, traveling at roughly 20 kilometers per second, and it struck the shallow sea covering what is now the Yucatan Peninsula of Mexico with a force so incomprehensibly large that the numbers required to describe it lose all meaning at the scale of human experience. The energy released by the impact has been estimated at roughly 100 million megatons of TNT, which is approximately 10,000 times the combined explosive yield of every nuclear weapon ever built. The rock that hit the ground was large enough that, if you stood on one side of it, you could not see the other side, and it was moving fast enough that, by the time anyone could have seen it in the sky, it would already have been too late for anything on Earth to do anything at all. The world was changed in seconds. It took millions of years for life to rebuild what was lost in that single morning. The immediate physical effects of the Chicxulub impact unfolded in a sequence of catastrophes that overlapped and compounded one another with terrible efficiency, beginning in the first seconds after contact and continuing for years and decades afterward. The initial impact vaporized much of the asteroid itself, along with billions of tons of the limestone and sulfur-rich rock of the Yucatan seafloor, launching a plume of superheated material into the upper atmosphere that spread around the entire planet within hours. Material ejected at high velocity re-entered the atmosphere at thousands of locations simultaneously, heating the air through friction as it fell and generating a global pulse of infrared radiation, intense enough to ignite wildfires across much of the planet's land surface. Geologists have found a layer of soot in rock formations of exactly this age on every continent, and that soot is the ash of forests that burned all at once, on every landmass, within days of the impact, releasing into the atmosphere the carbon that those forests had spent millions of years accumulating. The sky above a burning world turned black, and the light that life requires to sustain itself was cut off before the fires had fully cooled. The darkness that followed was not metaphorical. It was total, and it lasted long enough to end almost everything. Sulfur dioxide is a gas with the ability to form sulfate aerosols in the upper atmosphere, and these aerosols reflect incoming sunlight back into space with extraordinary efficiency, producing a cooling effect that can persist for years after a large volcanic eruption, or, as the Chicxulub impact demonstrated, after the vaporization of a sulfur-rich seafloor. The Yucatan target rock contained unusually high concentrations of sulfur-bearing minerals, and the impact vaporized an estimated 100 billion tons of sulfur dioxide in an instant, creating a reflective aerosol veil that climate models suggest would have reduced incoming solar radiation by as much as 80% for a period of 1 to 3 years following the impact. Without sunlight, photosynthesis collapses. Without photosynthesis, the base of almost every food web on land and in the oceans disappears within weeks. Large animals that require large quantities of food die first, because their energy reserves are exhausted before the smaller animals that require less. The herbivores go, and then the carnivores that depended on them. The collapse is not chaotic. It follows a brutal logic, proceeding from the largest and most food-dependent downward. And at every step of the collapse, it is the small, the adaptable, and the opportunistic that hold on longest. Evolution does not favor the mighty when the world goes dark. It favors the ones who can eat almost anything and survive on very little. The ocean did not escape the catastrophe of the Chicxulub impact, and its suffering was in some ways as profound as that of the land, though it played out through different mechanisms and at different speeds. The immediate physical effects included massive tsunamis generated by the impact itself, towering walls of water that traveled across the Gulf of Mexico and well into the interior of North America, depositing chaotic layers of mixed sediment that are still preserved in the geological record as evidence of a single catastrophic event. But the longer-term damage to the ocean came from chemistry rather than from waves, because the sulfur and carbon dioxide released by the impact began acidifying the ocean surface waters within weeks, and the calcium carbonate shells of the tiny marine plankton that formed the base of most ocean food webs began to dissolve in water that had suddenly become too acidic to support them. The collapse of marine plankton communities was effectively instantaneous in geological terms, and it propagated upward through every food web that depended on them, eliminating the ammonites, the mosasaurs, the plesiosaurs, and the marine reptiles that had dominated the oceans alongside the dinosaurs. The sea went quiet in a way that had not happened since the Great Permian extinction 250 million years before, and when life returned, it returned changed beyond recognition. Loss at this scale is not simply subtraction, it is transformation. Acid rain is a relatively mild environmental phenomenon in the modern world, associated primarily with industrial pollution and capable of damaging forests and acidifying lakes. But the acid rain that followed the Chicxulub impact was of an entirely different magnitude, generated by the combination of sulfur dioxide, nitrogen oxides, and water vapor in an atmosphere that had just been subjected to the most energetic single event in 66 million years. The nitrogen oxides were produced by the heating of atmospheric nitrogen and oxygen during the impact itself, a process that occurs whenever air is raised to extremely high temperatures, and these nitrogen oxides combined with atmospheric water vapor to produce nitric acid that fell back to Earth in rain across the entire planet. Combined with the sulfuric acid produced by the aerosols already in the atmosphere, This created precipitation that was orders of magnitude more acidic than anything modern industrial pollution produces, capable of stripping the waxy cuticle from leaves, killing sensitive plant tissues on contact, and further acidifying freshwater systems that were already under extreme stress from the loss of sunlight. The world that birds needed to survive was not simply dark and cold. It was also chemically hostile in ways that eliminated entire categories of plant and animal life, winnowing the survivors down to those with physiological and behavioral flexibility that none of them had been specifically evolved to possess. Survival in the aftermath of a catastrophe is the ultimate test of generalist ability. The duration of the impact winter, the period of reduced sunlight and lowered temperatures that followed the Chicxulub event, has been a matter of significant scientific debate, because the answer determines which animals had a realistic chance of surviving and which ones were effectively doomed regardless of their other adaptations. Climate models based on the estimated amount of material injected into the atmosphere suggest that a significant reduction in solar radiation persisted for somewhere between 1 and 3 years, with temperatures in continental interiors possibly dropping by as much as 15 to 20 degrees Celsius below normal during the peak of the cooling. This is not a trivial change. A drop of this magnitude is sufficient to eliminate frost-sensitive plants across entire latitudinal zones, to freeze shallow freshwater systems that would normally remain open year-round, and to disrupt the thermal environments that cold-blooded animals depend upon for their basic physiological functions. The birds that survived were almost certainly not surviving in comfort. They were surviving at the edge of what was physiologically and behaviorally possible for animals of their kind, exploiting every available food source, moving constantly in search of resources, and reproducing as fast as their biology allowed. The first few thousand years after the impact were not a time of rest. They were a time of relentless, desperate, daily survival. The birds that made it through did not do so because the world was kind to them. The KPG boundary, as geologists formally designate the layer of rock that marks the end of the Cretaceous and the beginning of the Paleogene period, is one of the most dramatic boundaries in the entire geological record, visible in rock outcrops around the world as a thin but unmistakable layer of clay enriched with the element iridium, which is rare in Earth's crust but relatively abundant in asteroids and other extraterrestrial objects. The discovery of this iridium anomaly by Lewis and Walter Alvarez in 1980 was one of the most consequential scientific findings of the 20th century, because it provided the first hard physical evidence that a large extraterrestrial impact had occurred at precisely the moment that the dinosaurs disappeared. The clay layer also contains shocked quartz, tiny crystals of silicon dioxide, whose internal structure has been deformed by pressures achievable only in asteroid impacts or nuclear explosions, and microscopic glass spherules produced by the rapid cooling of molten rock thrown into the atmosphere by the impact. Every one of these features tells the same story, and together they tell it with a precision that leaves no room for alternative interpretation. The geological boundary between the age of dinosaurs and the age of birds is not a gradual transition. It is a line, thin enough to cover with a thumbnail, that separates two completely different worlds. Everything changed on one side of that line. The animals that survived the Chicxulub extinction event and went on to diversify in the Paleogene were not random survivors. They were the beneficiaries of a specific set of biological traits that happened to confer survival advantages in the particular kind of catastrophe that had just occurred. And understanding those traits helps explain why birds made it through when so many other groups did not. Small body size was perhaps the most universally important survival trait, because small animals require less food per individual to survive, have shorter generation times that allow populations to recover more quickly from catastrophic losses, and can exploit food sources, such as seeds, insects, and detritus, that persist even when primary productivity has largely collapsed. Birds, which are on average substantially smaller than most of the large Cretaceous vertebrates that went extinct, benefited enormously from this size advantage. They also benefited from their dietary flexibility, their ability to fly and therefore to move across large distances in search of food, and from their capacity for rapid reproduction in favorable conditions. These traits did not evolve in anticipation of an asteroid strike. They evolved for entirely different reasons over tens of millions of years before the impact, and then they happened to be exactly the right combination of features for surviving the most unexpected catastrophe in the history of the vertebrate lineage. Fortune does not distribute itself according to merit, but sometimes the prepared mind, or the prepared body, is ready when fortune arrives. Seeds are among the most durable biological structures produced by any living organism, capable of remaining viable through conditions that would kill the parent plant outright, resistant to cold, drought, and darkness in ways that living plant tissue is not. And it was almost certainly this durability that made seeds the key resource sustaining the earliest bird survivors through the darkest and most food-depleted years of the post-impact world. Granivory, the consumption of seeds, is a feeding strategy found in a remarkably large proportion of modern bird species, and it is possible that the enormous diversity of seed-eating birds alive today is a direct legacy of the selection pressure imposed by the post-impact environment, which favored any animal that could exploit seeds as a primary food source. The birds that could crack open seeds, digest them efficiently, and find them buried in the soil or locked inside dried fruits had access to a food reserve that was largely inaccessible to reptiles and mammals that lacked the appropriate beak morphology or digestive chemistry. 66 million years later, a chicken scratching the ground for seeds in a farmyard is performing the same ancient behavior that its ancestors used to survive the end of the Cretaceous world, and the beak that does the work carries the shape of a solution to a problem that no longer exists. Old tools find new purposes. The form outlives the crisis that created it. The Paleocene epoch, which immediately followed the Cretaceous-Paleogene boundary and lasted from 66 to 56 million years ago, was a period of ecological reconstruction so fundamental that it essentially reset the template for vertebrate life on land, creating the conditions under which modern birds, mammals, and all the other groups that now dominate the terrestrial world first came into their ecological inheritance. The forests that regrew in the Paleocene were different from the forests of the Cretaceous, dominated by different plant families, structured differently in terms of light and shade, and the resources they offered to animals. Into these new forests, the surviving birds diversified with extraordinary rapidity, filling ecological roles that had been vacated by the extinction of so many non-avian dinosaurs and other Cretaceous animals. This process of ecological release, in which survivors diversify rapidly after a mass extinction, because so many formerly occupied niches have been emptied, is one of the most powerful forces in evolutionary history, and it drove the radiation of modern bird lineages with a speed that the fossil record of the Paleocene makes clearly visible. The first few million years after the extinction were, from an evolutionary standpoint, a period of explosive creative possibility, a blank canvas on which every survivor left its mark, and the birds painted more extensively and more durably than almost any other group. From catastrophe came creativity. From loss came the world we know. The survivors of the Chicxulub extinction event did not inherit a world of plenty. They inherited a world of ruins, and the first task of every survivor was simply to find enough food to stay alive from one day to the next, in an environment that had lost most of its primary productivity, most of its large animals, and most of the ecological relationships that had structured life for the previous 165 million years. The soil itself was transformed by the impact, because the loss of vegetation cover led to massive erosion, the disruption of soil microbial communities that normally cycle nutrients and maintain soil fertility, and the deposition across vast areas of a layer of impact debris that effectively buried the pre-impact soil surface beneath a sterile blanket of impact ejecta. Plants recolonized these disrupted soils in waves, beginning with spore producers like ferns, which are among the fastest colonizing plants and whose spore record in the immediate post-impact sediments shows a dramatic spike that paleobotanists call the fern spike, a signal of ecological catastrophe visible in the fossil record of virtually every continental region that has been sampled for post-impact vegetation. The birds that survived lived through and within this botanical recovery, adapting their diets and behaviors as the plant communities around them changed, and the flexibility that allowed them to track those changes was not incidental to their survival. It was the whole of it. Life after catastrophe is not about returning to what was. It is about learning what is now possible. The role of freshwater ecosystems in sustaining the post-impact survivors has received increasing attention from paleontologists in recent years, as studies of the relative survival rates of different animal groups across the KPG boundary have repeatedly found that freshwater-associated animals fared substantially better than their terrestrial counterparts. Turtles, crocodilians, freshwater fish, and many groups of aquatic invertebrates all passed through the extinction event with relatively low losses compared to terrestrial vertebrates. And the reason appears to be that freshwater food webs are more strongly buffered by detritus-based resources, dead organic matter that accumulates in sediments and can sustain aquatic communities, even when sunlight is severely reduced and primary photosynthesis has largely shut down. The birds that lived near freshwater, that could exploit aquatic insects, freshwater fish, and the detritus of aquatic ecosystems, had access to a food reserve that persisted through the impact winter more robustly than anything available in the devastated terrestrial environment. Many paleontologists now believe that the early surviving birds were water-associated, and that the richly diverse modern waterfowl and shorebird lineages may represent the groups that were closest to freshwater during the critical survival window. The edge of the water was, for a small feathered animal in the early Paleocene, the edge between life and death. The birds that found it lived. The birds that did not found something else, or found nothing at all. The Deccan Traps, a massive region of volcanic rock in what is now India, were formed by one of the largest episodes of volcanic activity in the past 100 million years, erupting over a period that overlapped with the Chicxulub impact event. And the relationship between these two catastrophic geological events and the end-Cretaceous extinction has been the subject of intense scientific debate for decades. Some scientists have argued that the Deccan volcanism was itself a significant contributor to the extinction, releasing enough carbon dioxide to warm the climate and enough sulfur dioxide to cool it intermittently across the final million years of the Cretaceous, potentially stressing ecosystems before the asteroid strike delivered the final blow. Others have argued that the impact itself may have triggered an intensification of the Deccan eruptions through the seismic energy it released, effectively compounding the biological damage of the impact by adding a sustained volcanic crisis on top of it. The question of which factor was more responsible, the impact or the volcanism, is probably the wrong question, because the evidence increasingly suggests that both were real, that both were consequential, and that the birds and other survivors had to navigate not just the aftermath of a single catastrophic impact, but an extended period of environmental instability driven by two overlapping geological crises. The survivors were not just lucky once. They were persistent enough to survive a world that was catastrophically unstable for much longer than any single event. The temperature record preserved in the chemistry of marine sediments from the KPG boundary shows not just a rapid cooling in the immediate aftermath of the impact, but a complex pattern of temperature changes extending across hundreds of thousands of years after the event, as different forcing mechanisms, the impact winter, the greenhouse gases from the Deccan volcanism, and the reorganization of ocean circulation all interacted in ways that produced a climate less stable than anything that had characterized the preceding Cretaceous. This instability was itself a biological filter, because animals with narrow thermal tolerances and highly specific habitat requirements were continuously challenged by a world that would not stay still long enough for them to adapt to its new baseline. The birds that survived were those with the broadest thermal tolerances, the widest habitat flexibility, and the most generalist behavioral repertoires, and these characteristics, selected for with brutal efficiency during the post-impact recovery period, became the foundation of the extraordinary ecological diversity of modern birds. The instability that could have destroyed them instead forged them, the way fire forges metal, by eliminating everything that was not flexible enough to survive the heat. What emerges from that forge is always something that could not have existed before it. The world made the birds it needed by destroying everything else. The chemical signature of the impact preserved in rock layers around the world has been analyzed in increasing detail as analytical techniques have improved. And what emerges from these analyses is a picture of a planet whose chemistry was fundamentally and rapidly altered across every major compartment: the atmosphere, the ocean, the soil, and the freshwater systems, in ways that persisted for far longer than the initial impact winter itself. Mercury, for example, which is a tracer for volcanic activity, shows elevated concentrations in KPG boundary sediments on multiple continents, consistent with the Deccan volcanism that was occurring simultaneously with the impact. Carbon isotope ratios show a sudden and dramatic shift at the boundary that records the collapse of primary productivity in the ocean, which is itself a proxy for the death of the photosynthetic base of marine food webs. Nitrogen isotope ratios record the disruption of the nitrogen cycle in soils. Sulfur isotopes record the input of volcanic and impact-derived sulfate into the atmosphere and ocean. Every element that participates in the chemistry of a living planet tells the same story from a slightly different angle, and together, they describe a world that was chemically as well as physically transformed by the events at the end of the Cretaceous. The birds that survived were breathing, eating, and drinking in a world whose chemistry had changed around them. And the fact that they managed to adapt to that changed chemistry is itself a testament to the remarkable physiological flexibility of the avian body. Chemistry changed. The birds changed with it. The psychological dimension of the post-impact survival period is not something the fossil record can directly address, because psychology leaves no bones, and we have no way of knowing what the inner experience of a small bird surviving the apocalypse actually felt like, if it felt like anything at all in a way that our concept of feeling could recognize. But we do know from modern behavioral research that birds are cognitively more sophisticated than was long assumed, that they demonstrate problem-solving, social learning, and in some species, what appears to be a rudimentary form of planning for future needs. And these cognitive capacities are not modern inventions in the bird lineage, but features inherited from the small-brained but behaviorally flexible theropods of the Cretaceous. The birds that navigated the post-impact landscape were doing so with brains that were capable of learning, of changing their behavior in response to novel circumstances, of exploiting new resources and avoiding new dangers in ways that a purely instinct-driven animal cannot. The cognitive flexibility that makes a modern crow capable of solving multi-step puzzles to obtain food is the same cognitive flexibility, expressed in a simpler and earlier form, that allowed the first Paleocene birds to find food in a world where all the old food sources had disappeared. Intelligence, or something near enough to it to make the difference, helped them survive. And the descendants of those survivors gave that intelligence to every bird alive today. The competition between birds and mammals for the ecological space, left open by the extinction of the non-avian dinosaurs, is one of the most fascinating and consequential biological contests in the history of life. And its outcome, the world we live in now, where mammals dominate the large-bodied terrestrial ecological niches and birds dominate the aerial ones while also filling many terrestrial niches of their own, was by no means a foregone conclusion in the early Paleocene. Both groups had survived the extinction event. Both were diversifying rapidly, and there were early experiments in which birds, rather than mammals, occupied the role of large terrestrial predator. The most dramatic of these experiments was the evolution of the terror birds, formerly called phorusrhacids, a group of large, flightless, carnivorous birds that dominated the predator guild in South America for tens of millions of years while that continent remained isolated from North America. Some terror birds stood more than 2 meters tall and possessed beaks powerful enough to crack bone, and they were the apex predators of their ecosystems, in a way that made them functionally equivalent to the large theropod dinosaurs whose ecological role they had, in a sense, inherited. The terror birds eventually went extinct when the land bridge connecting North and South America was established, and mammalian predators entered from the north. But for tens of millions of years, they demonstrated that the bird body plan was fully capable of occupying the very ecological space once held by the dinosaurs. Birds tried to be dinosaurs again, and for a while they succeeded. The isotope chemistry of early Paleocene bird bones has been analyzed in several studies to reconstruct the dietary habits of the earliest post-extinction bird communities. And what these analyses reveal is a picture of remarkable dietary flexibility across the surviving lineages, with different groups exploiting seeds, insects, freshwater invertebrates, fish, carrion, and plant material across a range of combinations that reflects both the scarcity of any single food source and the opportunism that characterized survival in the post-impact world. Stable isotopes of carbon and nitrogen incorporated into bone during growth carry signatures that reflect the diet of the animal. And by comparing these signatures to those of known food sources in the same geological deposits, paleontologists can reconstruct the approximate dietary preferences of extinct animals with a precision that was simply not possible before isotope geochemistry became a standard analytical tool. What these studies confirm is that the early bird survivors were not specialists. They were generalists of the most extreme kind, eating whatever was available, switching between food sources as conditions changed, and maintaining populations across a wide range of habitat types, rather than restricting themselves to the specific ecological niches they might have occupied in the more structured world of the late Cretaceous. Generalism is not a glamorous survival strategy. It does not produce the most spectacular or the most beautifully adapted animals, but it produces animals that survive, and survival is its own kind of excellence. Now let the weight of that great silence after the impact rest upon you as gently as ash falls on still water. The world grew very quiet after the catastrophe, and in that quiet the small survivors moved carefully through a changed landscape, finding what they could, eating what they found, living one day at a time in a world that was still deciding what it wanted to become. Your breath mirrors that quiet now, slow and even and without urgency, and the darkness behind your eyes is not the darkness of that ancient winter but something softer, something that belongs to you and to this moment, and to the easy passage from waking into sleep. The birds found their way through. You are here because they did. Let that be enough to carry you forward into rest. Archaeopteryx is the most famous transitional fossil in the entire history of paleontology, and it earned that fame not through any single dramatic feature, but through the way it combined, in one small body preserved in the fine-grained limestone of a Bavarian quarry, the anatomical characteristics of both non-avian dinosaurs and modern birds, in a proportion that made the evolutionary relationship between the two groups impossible to deny, once the specimen was examined carefully. The first Archaeopteryx specimen was discovered in 1861, just 2 years after Charles Darwin published On the Origin of Species, and its timing was so convenient for the new theory of evolution that some early observers suspected it might be a forgery. It was not a forgery. It was something far more interesting, a genuine animal that had lived approximately 150 million years ago in a shallow tropical archipelago that would one day become southern Germany, an animal with the feathered wings and wishbone of a bird, and the toothed jaws, clawed fingers, and long bony tail of a theropod dinosaur. For more than a century, Archaeopteryx was considered the first bird, the definitive point at which the dinosaur lineage crossed the threshold into avian identity. Modern science has complicated this picture considerably, revealing a much richer and more tangled evolutionary transition than any single transitional fossil can represent. But Archaeopteryx remains the most eloquent single embodiment of the moment when the sky opened its doors to a new kind of animal. Some thresholds leave marks in stone. The question of how flight evolved in birds has generated one of the longest and most productive scientific debates in the history of paleontology, with two major competing hypotheses that have traded scientific favor back and forth across more than a century of research. The first hypothesis, known informally as trees down, proposes that the ancestors of birds were arboreal animals that glided or parachuted from trees and gradually evolved powered flight from the gliding condition, a sequence that has the aerodynamic advantage of allowing gravity to assist in the early stages of flight evolution. The second hypothesis, known as ground up, proposes that the ancestors of birds were cursorial, ground-running animals that evolved flight by first developing the ability to generate thrust and lift during fast running, with the wings initially functioning as aids to running on inclined surfaces rather than as flight surfaces proper. A third possibility, that both mechanisms contributed at different stages in different lineages, has gained support in recent years as the fossil evidence from China has revealed a far more diverse array of early feathered theropods than was previously known, some of which appear to have been arboreal, and some of which were almost certainly terrestrial. Evolution rarely chooses between two options when it can find a way to explore both simultaneously, and the origin of flight may turn out to be not a single event, but a convergent achievement reached by multiple lineages through multiple paths. The sky was not won in one attempt. It was discovered again and again. The wishbone, or furcula, is one of the most recognizable anatomical features of a modern bird, a forked bone formed by the fusion of the two clavicles that functions as a spring during the flight stroke, storing and releasing elastic energy with each beat of the wing, and contributing to the efficiency of powered flight. The discovery that wishbones are not unique to birds, but are present in many non-avian theropods, including Tyrannosaurus rex and Velociraptor, was one of the landmark findings of late 20th century paleontology, because it demonstrated that this key avian feature evolved well before flight itself, in animals that were certainly not flying, for purposes that must have had nothing to do with aerial locomotion. In non-avian theropods, the furcula appears to have functioned as a structural element of the shoulder girdle, possibly contributing to the mechanics of the prey-catching strike, and its recruitment into a flight-related role in the avian lineage is a clear example of exaptation, the evolutionary process by which a structure evolved for one function is co-opted for a completely different one. The wishbone in a roasted chicken that two people pull apart at a dinner table is the same structure that helped T-Rex hold struggling prey, repurposed across 66 million years of evolution into something that lifts rather than grabs, that gives rather than takes. The same bone, a completely different story. The coevolution of birds and flowering plants is one of the great collaborative stories in the history of life, a mutually beneficial relationship that transformed both groups simultaneously and produced much of the biological richness that characterizes the modern world. Flowering plants, or angiosperms, began their diversification during the Cretaceous period, and they diversified in concert with the insects, birds, and mammals that pollinated their flowers, dispersed their seeds, and were in turn fed by their fruits and seeds across hundreds of millions of years of intimate ecological interaction. Birds, with their mobility, their ability to travel long distances between feeding sites, and their tendency to deposit seeds in their droppings far from where they consume the parent fruit, became among the most important seed dispersers for a vast number of plant species. And many flowering plants evolved fleshy, brightly colored fruits, specifically adapted to attract birds. With colors in the red and orange range that are highly visible to avian tetrachromatic vision, and with sizes and shapes calibrated to the beak dimensions and swallowing capacity of particular bird species. The relationship between a bird and the fruit it eats is not a simple transaction. It is the current expression of a negotiation that has been ongoing for tens of millions of years, in which both parties have been continuously shaped by the other, and the chicken that pecks at fallen fruit in a farmyard is participating, unknowingly, in one of the oldest and most consequential partnerships in the history of life on Earth. We are all embedded in relationships older than we can imagine. The diversity of beak shapes in modern birds is one of the most striking and well-studied examples of adaptive radiation in the entire animal kingdom, a demonstration of how a single basic anatomical structure can be modified through natural selection into forms as different as the massive crushing bill of a parrot, the long probing needle of a hummingbird, the flat sieve of a flamingo, and the hooked tearing weapon of an eagle. This diversity of beak form did not arise all at once. It accumulated over tens of millions of years of diversification following the extinction of the non-avian dinosaurs, as bird lineages colonized different ecological niches and evolved beak morphologies precisely calibrated to the food sources available in those niches. The developmental genetics underlying beak shape variation have been studied in remarkable detail in recent years, particularly through the work of researchers studying the famous Darwin's finches of the Galapagos Islands. And what these studies have revealed is that the entire spectrum of beak shape variation in birds can be produced by adjustments in the expression of a surprisingly small number of developmental genes, particularly those controlling the growth and patterning of the facial skeleton during embryonic development. Small changes in a few genes, timed and placed with precision during embryogenesis, produce beaks as different as a toucan's and a sparrow's, and these same developmental mechanisms were present, in a simpler form, in the theropod ancestors of birds long before the beak itself had fully evolved. The diversity of the world is built from a small vocabulary of developmental instructions, spoken in different combinations in every living thing. The evolution of endothermy, or warm-bloodedness, in the bird lineage is a story that extends deep into the theropod ancestry of birds and involves not a single evolutionary transition, but a gradual accumulation of physiological changes across tens of millions of years that collectively produce the high-performance metabolic systems of modern birds. The evidence for this gradual escalation comes from multiple sources, including the oxygen isotope chemistry of fossil bone, which records body temperature during growth, the growth rate evidence already discussed, and the discovery of insulating feather-like structures in theropods that predate the origin of birds by many tens of millions of years. Modern birds have metabolic rates substantially higher than those of mammals of similar body size, and they maintain body temperatures typically between 40 and 42 degrees Celsius, higher than the normal human body temperature of 37 degrees. This extreme thermal intensity is not a coincidence. It is connected to the enormous energy demands of flight. which requires sustained muscular output at levels impossible to achieve without the constant delivery of oxygen and metabolic fuel that only a high-powered, warm-blooded physiology can provide. The body temperature of a chicken, taken by a veterinarian and registered at 41 degrees, is the same extraordinary metabolic fire that powered the first feathered animals to lift themselves into the sky, inherited through an unbroken chain of warm bodies stretching back into the Cretaceous forests, where the story of birds truly begins. The evolution of the avian egg, one of the most perfectly engineered biological structures ever produced by natural selection, represents a thread of continuity connecting modern birds directly to their dinosaurian ancestors in a way that is both scientifically profound and visually immediate. Dinosaur eggs have been found in extraordinary abundance and diversity in fossil deposits around the world, and their structural characteristics, the microscopic architecture of the shell, the arrangement of the pores that allow gas exchange, the shape and proportions of the overall egg, show a clear progression from more reptilian forms in early dinosaurs to increasingly bird-like forms in the theropod lineages most closely related to birds. The eggs of Oviraptorosaurs, a group of theropods closely related to birds, are structurally nearly identical to the eggs of modern rat-type birds like emus and ostriches, with the same basic shell microstructure, the same pore arrangement, and the same asymmetric shape. The eggshell that a chicken produces in the span of 24 hours, laying down calcium carbonate crystal in a precise architectural pattern controlled by proteins that have been conserved across hundreds of millions of years of vertebrate evolution, is a biological document of enormous antiquity, a structure whose basic design was settled long before the first true bird ever existed. When you crack open a morning egg, you are holding in your hands the product of 300 million years of evolutionary refinement. The shell is ancient. The protein that built it is older still. The feather follicle, the small pocket of specialized skin tissue from which every feather grows, is one of the most important and least appreciated structures in the biology of birds, because it is the developmental engine that produces the entire feather, from the simplest down feather of a nestling to the most elaborate tail plume of a peacock. And it achieves this through a precisely choreographed sequence of molecular events that has been conserved with remarkable fidelity across the entire evolutionary history of the bird lineage. The genetic programs that control feather development have been studied intensively in recent years, particularly in the context of understanding the evolutionary origin of feathers. And what this research has revealed is that the feather follicle is a highly derived structure that evolved from the simpler scale follicle of the ancestral reptile through a series of modifications in the regulatory networks controlling skin development. Genes that in reptiles control the formation of scales were gradually co-opted and modified to produce increasingly complex epidermal outgrowths, beginning with simple filaments and progressing through branched filaments to the fully asymmetric, aerodynamically optimized flight feathers of modern birds, across a developmental evolutionary trajectory that is still partially visible in the development of individual feathers within the follicle of a living bird. The feather that grows from the wing of a chicken recapitulates, in its individual development, a compressed version of the evolutionary history of feathers themselves. The personal and the historical collapse into each other in every follicle. The lung of a modern bird is a fixed-volume organ that does not expand and contract with each breath the way a mammalian lung does. And this structural difference is the key to understanding why the avian respiratory system is so much more efficient than our own, and why its evolutionary origins in the non-avian theropods gave those animals a physiological advantage that helped drive their ecological dominance across the Cretaceous period. In the bird respiratory system, air flows in through the trachea and into a set of posterior air sacs, which function as bellows to push air through the parabronchi, the gas exchange surfaces of the fixed lung, and then out through a set of anterior air sacs before being expelled. This unidirectional flow means that the parabronchi are always bathed in fresh, oxygenated air rather than in the mixture of fresh and stale air that passes through the mammalian lung in both directions. The result is a gas exchange efficiency that allows birds to extract oxygen from air at altitudes and under exercise conditions that would be physiologically impossible for a mammal of similar body size. Bar-headed geese migrate over the Himalayas at altitudes that would render a human unconscious within minutes, sustained by a respiratory system whose basic architecture was laid down in the theropod ancestors of birds tens of millions of years before any bird attempted a mountain crossing. The air sacs of a chicken, invisible inside its body, are the architectural descendants of structures that once sustained animals running across the Cretaceous plains. The breath connects everything. The immune system of modern birds shows a fascinating mixture of ancient conserved elements, shared with all vertebrates, and derived features that appear to be specific to the avian lineage, or to the broader archosaur group that includes birds and crocodilians. Among the most distinctive features of the avian immune system is the bursa of Fabricius, a specialized lymphoid organ unique to birds that is responsible for the maturation of B cells, the immune cells that produce antibodies, and which gives B cells their name, the B standing for bursa rather than for bone marrow, as is sometimes assumed. The bursa of Fabricius has no direct equivalent in any other vertebrate group, suggesting that it evolved specifically within the bird lineage as a novel immunological specialization, possibly in response to the particular infectious challenges faced by animals living in close proximity, with conspecifics in nesting colonies or roosting groups. The conservation of immune system architecture across the bird lineage is also striking, with the basic organization of the avian immune response remaining recognizable across species as divergent as ostriches, penguins, and sparrows, suggesting that it was established early in avian evolution and has been highly resistant to subsequent modification. When a chicken mounts an immune response to a pathogen, it does so using cellular and molecular machinery that has been refined across approximately 100 million years of avian evolution. Machinery that was already ancient by the time the first domestic chicken was kept by a human hand. The colonization of oceanic islands by birds is one of the clearest and most studied examples of evolution in action, and it provides a window into the evolutionary processes that shaped early bird diversification in the Paleogene, just as vividly as it illuminates the more recent radiations that Darwin and his contemporaries first began to document. When birds colonize a remote oceanic island, they arrive in an environment that lacks most of the competitors, predators, and parasites that constrain their evolutionary options on their continent of origin. And they respond to this ecological release by diversifying in body size, diet, behavior, and sometimes even locomotion. With some island bird lineages independently evolving flightlessness, when the absence of ground predators makes the energetic cost of maintaining flight musculature unjustifiable. This pattern of island colonization followed by rapid diversification is a small-scale replay of the process by which the ancestors of modern birds diversified across the post-extinction Paleogene world, colonizing the ecologically empty landscape left by the extinction of the non-avian dinosaurs, and diversifying into every available niche with a speed and thoroughness that the fossil record makes startlingly clear. The same evolutionary dynamics that produce a new species of honeyeater on a Pacific island over 10,000 years produced the entire diversity of modern birds over 66 million years. And the principles are identical in both cases, because evolution operates by the same rules at every scale. The island and the continent differ only in size and time. The creativity is the same. The bone microstructure of early Paleogene birds, examined in thin section under polarized light, reveals a pattern of rapid and sustained growth that stands in sharp contrast to the slow, episodic growth patterns of most living reptiles and resembles instead the fast, fibrillamular bone growth of modern birds and mammals, confirming that the high growth rate physiology that characterizes living birds was already established in the earliest members of the modern bird radiation, rather than being a later acquisition. This matters for understanding the evolutionary trajectory of the post-extinction bird diversification, because high growth rates require high metabolic rates, and high metabolic rates confer the ability to remain active across a wider range of temperatures, to sustain higher levels of locomotor activity, and to support faster reproductive cycles, all of which would have been significant advantages in the unstable, resource-limited environment of the early Paleocene. The bone of an early Paleogene bird is not merely a structural scaffold. It is a record of the animal's physiology inscribed in mineral form, readable by anyone trained to interpret its microscopic architecture. And what it says is that the birds of the early Paleocene were already fully committed to the high-energy, high-performance physiological strategy that makes modern birds the most diverse and widespread group of terrestrial vertebrates on the planet. The bones knew what kind of animal they were building long before paleontologists learned to read the message they contained. The evolution of bird song and vocalization represents one of the most complex behavioral systems in the animal kingdom, a cultural as well as biological inheritance in which learned vocalizations are transmitted from generation to generation, alongside the genetic endowment that makes learning possible, creating a dual inheritance system that has no parallel in most other animal groups. The neural substrate of song learning in birds, studied in extraordinary detail in the past 50 years of neuroscience research, involves a set of specialized brain nuclei, collectively known as the song system, a circuit dedicated to the learning, storage, and production of complex vocalizations that has no exact equivalent in any other vertebrate brain, though homologous regions exist. The song system develops under the influence of sex hormones during a critical period in early development, during which young birds are capable of learning the vocalizations of their species by listening to adults. And this period of hormone-sensitive social learning is itself a derived feature of the avian lineage that was not present in the non-avian dinosaur ancestors of birds. Yet the vocal apparatus that produces bird song, the syrinx, a structure unique to birds located at the junction of the trachea and bronchi, may have had precursors in the non-avian theropods, and there is ongoing research into whether some dinosaurs were capable of producing complex vocalizations that contributed to their social communication. The song of a bird in the morning is not only music. It is the latest iteration of a communication system that has been evolving for longer than our species has existed. The geographical pattern of modern bird diversity, with the highest species richness concentrated in the tropical regions of South America, Africa, and Southeast Asia, and declining toward the poles and toward drier inland regions, reflects a combination of evolutionary history, current climate, and the productivity of different ecosystems. And its origins can be traced back to the early Paleogene diversification of the modern bird lineages in the equatorial regions that remained most biologically productive through the extinction event and its immediate aftermath. The tropics served as refugia, regions where the biological impact of the extinction was relatively less severe, because productivity was maintained at somewhat higher levels by the higher solar radiation received at low latitudes, and the bird lineages that diversified most extensively in these refugia seeded the modern tropical bird diversity that researchers and birdwatchers marvel at today. The temperate and polar bird faunas are in large part derived from more recent colonization events by lineages that originated in the tropics, adapting their physiology and behavior to the more seasonal and climatically challenging environments of higher latitudes as conditions changed and new habitats became available. A chicken, descended from the red junglefowl of the tropical forests of Southeast Asia, carries in its genome and its behavior the heritage of that tropical origin, the preference for dense vegetation, the sensitivity to cold, the foraging strategy calibrated to a world of leaf litter and fallen fruit rather than open fields and managed grain stores. The farmyard is not the jungle, but the chicken has not entirely forgotten where it came from. The discovery that birds are, in the most technically precise sense of the term, living dinosaurs, is not merely a semantic reclassification, but a fundamental reconceptualization of what an animal is and what its evolutionary heritage means for its biology. The modern cladistic classification of organisms, which groups animals strictly according to their evolutionary relationships rather than their overall similarity of appearance, places birds within the Dinosauria, specifically within the theropod subgroup, as the only surviving lineage of that once enormously diverse group. This classification means that the statement "we have no living dinosaurs" is literally false, that every bird alive today is a dinosaur in the same sense that a bat is a mammal or a gecko is a reptile, and that the extinction of the non-avian dinosaurs 66 million years ago was not the extinction of dinosaurs, but the extinction of the non-avian members of the group, leaving the avian members to continue the lineage into the present. There are approximately 10,000 living species of birds, distributed across every continent and nearly every habitat on Earth, making the surviving dinosaur lineage one of the most species-rich groups of terrestrial vertebrates alive today. The dinosaurs did not go extinct. They went small and grew feathers, and eventually one of them ended up in your kitchen producing eggs for your breakfast. The story did not end 66 million years ago. It simply changed its shape entirely. The rate of molecular evolution in birds, measured by comparing the DNA sequences of species whose divergence times are known from the fossil record, has been estimated with increasing precision as genomic sequencing has become faster and cheaper. And the results have implications not just for our understanding of bird evolution, but for the broader science of molecular clocks. The technique of using rates of genetic change to estimate divergence times when fossil evidence is absent or incomplete. Bird genomes, which are among the smallest and most compact of any vertebrate group, with less repetitive DNA and fewer transposable elements than mammalian genomes of similar gene content, evolve at a rate that is slower in some respects than mammalian genomes and faster in others. And disentangling these different rates has been one of the technical challenges of avian molecular evolution. The small genome size of birds is itself an adaptation connected to flight, because smaller cells can be perfumed with oxygen more efficiently, and reducing genome size, which reduces cell size, may have been selected for as an indirect consequence of the metabolic demands of powered flight. The compactness of the chicken genome, studied in extraordinary detail since it was first sequenced in 2004, is therefore not simply a feature of chickens, but a feature of birds generally, connected by an evolutionary chain of causation to the demands of an aerial lifestyle that the chicken itself no longer pursues. The genome was shaped by the sky, even in an animal that no longer flies toward it. The biogeography of early bird evolution in the Paleogene has been reconstructed from a combination of fossil evidence, molecular phylogenetics, and the comparative anatomy of living bird groups. And the picture that emerges is one of extraordinary dynamism, with lineages dispersing across oceans, colonizing new continents, and diversifying rapidly in isolation before reconnecting with their relatives when continental configurations changed. The ratites, the group of large flightless birds that includes ostriches, emus, rheas, cassowaries, and kiwis, were once thought to represent a single ancient lineage that was distributed across the supercontinent Gondwana before its breakup separated the different lineages onto different continents. But modern molecular analysis has revealed that the ratites evolved flightlessness multiple times independently from flying ancestors that dispersed across the oceans. A finding that fundamentally revised our understanding of how large flightless birds came to be present on so many different southern hemisphere landmasses. The kiwi, now confined to New Zealand, has relatives whose ancestors flew across the Pacific from Australia relatively recently in evolutionary terms, not ancient survivors of a sunken Gondwanan land bridge. The world of birds is full of surprises of this kind, discoveries that reveal how wrong our intuitions about evolutionary history can be when we fail to take seriously the capacity of small animals to cross very large distances. The ocean is not always a barrier. Sometimes it is a highway if you have wings. Let the early Paleogene world settle around you now like the settling of new sediment on an ancient ocean floor, each particle drifting down slowly in the dark water, finding its place without urgency, without effort, simply following the quiet pull of gravity toward rest. The proto-birds have found their way through the darkness, and the forests are beginning to grow again. And somewhere in the green recovering world, a small feathered animal is singing a song that no human ear has ever heard, a song whose descendants will one day be heard on every continent. Your body is heavy in the way that a body becomes heavy when it finally lets go, and the thoughts that have been moving through you are slowing now, taking longer to arriving softer. The story continues while you rest. You do not need to follow every step of it. The birds know the way. The chicken, known to science as Gallus domesticus, is descended from the red junglefowl of the tropical forests of South and Southeast Asia, and its domestication by human societies represents one of the most consequential biological partnerships in the history of our species, producing an animal so thoroughly integrated into human food systems that there are now more chickens alive on Earth at any given moment than there are humans, with estimates placing the global chicken population at approximately 25 to 30 billion individuals at any one time. The red junglefowl itself is a member of the pheasant family, the Phasianidae, a group with deep evolutionary roots extending back into the early Paleogene diversification of the modern bird orders, and its relationship to the broader avian family tree places it firmly within the lineage that emerged from the KPG extinction and diversified across the recovering Paleogene world. But the chicken is not merely an interesting domesticated bird. It is, in the context of the scientific story we have been tracing, the most studied, most accessible, and most genomically characterized living representative of a lineage that connects directly and unbroken to the non-avian dinosaurs of the Cretaceous period. Every biological feature of the chicken, every protein in its cells, every regulatory sequence in its genome, every structural detail of its bones and feathers, is a piece of evidence in the ongoing scientific reconstruction of what dinosaurs were actually like. The most common bird in the world is also, in a very real sense, the most informative dinosaur fossil that has ever been found. It is just that this fossil is still alive. The genome of the chicken was first sequenced and published in 2004, making it the first bird and the first agricultural animal to have its complete genetic blueprint decoded. And the information contained in that sequence has transformed our understanding not just of chicken biology, but of vertebrate evolution more broadly. By providing a reference point from which the evolutionary distances between different lineages could be precisely measured, and the timing of key evolutionary events could be estimated with unprecedented accuracy. The chicken genome contains approximately 1 billion base pairs of DNA, organized into 39 pairs of chromosomes, which is substantially smaller than the human genome of 3 billion base pairs, despite the chicken containing a similar number of protein-coding genes, roughly 15 to 17,000. This compactness reflects the general tendency of bird genomes toward economy and compression, with less repetitive DNA, fewer transposable elements, and more tightly packed gene arrangements than mammalian genomes of comparable gene content. Comparing the chicken genome to the genomes of other vertebrates has revealed the deep conservation of many gene sequences across hundreds of millions of years of evolution, with some regulatory regions of the chicken genome bearing recognizable similarity to equivalent regions in the genomes of fish, amphibians, and even invertebrates, demonstrating that the molecular toolkit underlying vertebrate development was assembled very early in animal evolution and has been modified but not fundamentally rebuilt across the entire subsequent history of the group. The genome of a chicken is a library of evolutionary history, most of whose books have never been read. The protein collagen, which is the most abundant structural protein in the bodies of all vertebrates, provides one of the most direct and surprising molecular connections between chickens and non-avian dinosaurs, because collagen molecules are durable enough under certain exceptional preservation conditions to survive in fossil bone for tens of millions of years. And when they do survive, their amino acid sequences carry evolutionary information that can be used to place the animal that produced them in its correct position in the vertebrate family tree. In 2007, a research team led by Mary Schweitzer reported the recovery of collagen peptide sequences from a Tyrannosaurus rex specimen, approximately 68 million years old. And when those sequences were compared to the collagen sequences of living animals, the closest match was not to any reptile, but to the collagen of modern birds, specifically to the collagen of chickens. This finding, controversial when first published and subsequently replicated and extended by additional studies, provided the first direct molecular evidence connecting the protein chemistry of T. rex to that of modern birds. And it did so not through inference or analogy, but through the actual comparison of amino acid sequences separated by 68 million years of time. The collagen in the bones of a chicken is, in its deepest molecular structure, the same protein as the collagen that held together the skeleton of the largest predator of the Cretaceous. The material and the monster share their most fundamental architecture. The discovery of soft tissue preservation in dinosaur bones by Mary Schweitzer and her colleagues at North Carolina State University is one of the most controversial and ultimately consequential findings in the history of paleontology, challenging deeply held assumptions about the maximum possible duration of biological molecule survival in the fossil record, and opening an entirely new avenue of inquiry into the molecular biology of extinct animals. Schweitzer first reported the observation in 2005, when she dissolved the mineral matrix from a T. rex femur bone and found flexible, translucent structures that retain the microscopic morphology of blood vessels, cells, and connective tissue fibers. Initial skepticism was intense, because the accepted understanding of organic molecule degradation rates suggested that biological structures of this kind should be completely destroyed over millions of years by hydrolysis, oxidation, and microbial decomposition. Subsequent research identified the preserved structures as containing not just collagen, but also what appear to be remnants of cellular structures, possibly including fragments of the original dinosaur cells themselves, though the interpretation of these structures remains actively debated. The mechanism that allows such preservation is still not fully understood, though iron from blood hemoglobin has been proposed as a cross-linking and preservation agent. What is not debated is the implication that the boundary between the living and the fossilized, between the biological and the geological, is more permeable than anyone had previously believed. The past is more present than it appears. The scales on the legs and feet of a chicken are not simply a remnant of the reptilian ancestry of birds in the general sense, but are more specifically the product of a specific developmental genetic switch that suppresses the full feather developmental program and reverts the skin of the lower leg to the production of a simpler, scale-like epidermal appendage. Research into the developmental genetics of feathers and scales in birds has demonstrated that the genes responsible for feather development, including signaling molecules like sonic hedgehog and bone morphogenetic proteins, are active across the entire skin of the embryonic bird, and that the distinction between feathered skin on the body and scaled skin on the legs arises from the differential suppression of parts of the feather developmental pathway in the leg epidermis specifically. When this suppression is removed experimentally, by manipulating the expression of specific developmental genes in chicken embryos, the leg scales are replaced by feather-like structures, a finding that directly demonstrates that the scales of modern birds are developmentally derived from the feather program, rather than representing a more ancient and separate structure. This relationship means that the scaly leg of a chicken is not a window into a pre-feathered past, but rather a derived condition, a feather that has been switched off, and the switching mechanism itself is something that can be found in the regulatory genome of every chicken alive, a set of molecular instructions that says, here, in this part of the body, do not make a feather, make what we used to make before. The past is encoded as an option that development can choose. The wing of a chicken contains, embedded within its feathers, an underlying musculature and bone, vestigial claw digits, that represent the evolutionary remnants of the three-fingered hand of the theropod ancestor, reduced but not eliminated by millions of years of selection, that progressively transformed the grasping hand into the flight-enabling wing. In adult chickens, these vestigial claws are small and largely non-functional, present at the leading edge of the wing as tiny curved structures that the bird does not use for any apparent purpose in its daily life. But in the embryos of chickens and other birds, these digital remnants pass through a developmental stage in which they are more prominent and more clearly digit-like, before being partly resorbed and reduced to their adult form. And the developmental sequence captures in miniature the evolutionary history of the avian forelimb, each embryo recapitulating in days the transformation that took millions of years to establish in the adult morphology of the lineage. In some bird species, particularly among the ratites and certain other groups, the wing claws are more prominent than in chickens, and in the Watsine, a South American bird whose chicks use prominent wing claws to clamber through vegetation before they are capable of flight. The functional use of wing claws in juveniles provides a glimpse of the ecological context in which such structures might have been maintained by selection during the early evolution of birds. The chicken's vestigial claws are not mistakes or oversights. They are memories, kept just in case the world should change again and find a use for them. The thyroid hormone system of modern birds, which plays a central role in regulating metabolism, growth, development, and the timing of seasonal behaviors including migration and molt, has deep evolutionary roots that extend back through the amniote lineage to ancestors shared with modern reptiles. And the comparative study of this hormonal system across birds, crocodilians, and other reptiles has provided insights into how metabolic regulation evolved in the archosaur lineage that includes both birds and their non-avian dinosaur relatives. Thyroid hormones, specifically thyroxine and triiodothyronine, are produced by the thyroid gland and act on virtually every cell in the body, regulating the rate of oxygen consumption, heat production, and the expression of many genes involved in development and differentiation. In birds, the thyroid system is particularly active during periods of high metabolic demand, including migration, breeding, and molting, and the sensitivity of the avian thyroid system to environmental cues like day length and temperature allows birds to synchronize their physiology with the seasonal rhythms of their environment with remarkable precision. The thyroid hormone receptors that mediate these effects in the cells of a chicken are molecular structures whose basic architecture has been conserved across more than 300 million years of tetrapod evolution. Modified in their details but recognizable in their fundamentals across every vertebrate lineage that has been examined. The same molecular lever that governs the metabolism of a chicken governed, in some related form, the metabolism of the animals that gave rise to it. The controls are ancient. Only the settings have changed. The skeletal similarities between theropod dinosaurs and modern birds extend to a level of detail that is genuinely startling when examined carefully. Because the correspondence is not merely superficial resemblance but a bone-by-bone homology in which specific bones in specific positions perform equivalent mechanical functions in animals separated by 66 million years of evolution. The ankle joint of a bird, for example, is a modified version of the ankle joint found in theropod dinosaurs, with the same basic arrangement of bones but modified in its proportions and angles to suit the demands of bipedal locomotion in an animal whose body is oriented differently from its dinosaurian ancestor. The pubic bone of a bird, which in most theropods points forward as is typical of the lizard-hipped saurischian condition, has rotated backward over the course of avian evolution to a position more similar to the bird-hipped ornithischians, and this rotation is actually observable in embryonic development, where the pubic bone begins in a more ancestral orientation and rotates to its adult position during development. Another case of embryonic recapitulation of evolutionary history. When an anatomist studies the skeleton of a chicken and the skeleton of a small theropod dinosaur side by side, the similarities are not the impressionistic similarities of two things that generally resemble each other. They are the precise correspondences of structures derived from a common source, modified by time and selection, but carrying the unmistakable signature of shared origin. The bones remember where they came from. The protein keratin, which forms the structural basis of feathers, scales, claws, beaks, and the outer layer of skin in all birds and reptiles, exists in two major forms in vertebrates: the alpha-keratins found in all vertebrates and the beta-keratins found only in reptiles and birds. And the evolutionary history of the beta-keratin gene family provides a clear molecular record of the evolutionary relationships among the groups that possess it. In birds, the beta-keratin genes have undergone an extensive expansion compared to the ancestral reptilian condition, with the chicken genome containing dozens of beta-keratin genes organized in clusters, each cluster corresponding to a different structural context, with feather keratins, scale keratins, claw keratins, and beak keratins, each encoded by a distinct subset of genes that are expressed in the appropriate tissue during development. The feather keratins, which are the most numerous and most structurally complex of the bird beta-keratins, appear to have evolved from scale keratins through a series of gene duplication and divergence events that are partially reconstructible from the sequence relationships among the different keratin genes in the chicken genome. This means that the molecular material from which feathers are built is chemically related to the molecular material from which scales are built, and the evolutionary transition from scale to feather is readable, at the molecular level, in the family tree of the keratin genes themselves. The chemistry of a feather is the chemistry of a scale that has been through an experience and come out transformed. Chemistry, like anatomy, carries its history forward. The hemoglobin of birds is a variant of the hemoglobin found in all vertebrates, built from the same basic globin protein subunits that carry oxygen in the blood of fish, amphibians, reptiles, and mammals, but modified in its oxygen-binding properties in ways that are precisely calibrated to the metabolic demands and physiological conditions experienced by flying animals at altitude. Avian hemoglobin has a higher affinity for oxygen at lower partial pressures than mammalian hemoglobin, allowing birds to load their blood with oxygen, even in the thin air at high altitude. And this property is achieved through specific amino acid substitutions at particular positions in the globin protein chain that alter the cooperative binding behavior of the hemoglobin tetramer. These amino acid substitutions are not random. They are precisely positioned at sites known from structural studies to influence oxygen affinity, and their presence in bird hemoglobin and absence in the hemoglobins of non-flying reptiles demonstrates that they were selected for specifically in the avian lineage, in connection with the evolution of powered flight and the metabolic demands it imposes. The globin genes from which bird hemoglobin is built are the same genes, in evolutionary terms, as those that build human hemoglobin, diverged from a common ancestor in the Devonian period more than 350 million years ago, and modified continuously in both lineages, since then, in response to the different physiological demands of different ways of life. The oxygen that flows through a chicken's blood is carried by a molecule whose evolutionary history predates the dinosaurs by nearly 200 million years. The blood is old. The molecule is older still. The nervous system of the chicken, while considerably less complex in its overall organization than the nervous system of a mammal of similar body size, contains within its structure a set of features that are diagnostic of the archosaur lineage, and that connect the chicken's neurobiology directly to the neurobiology of its dinosaurian ancestors. The cerebellum of the chicken, the brain region responsible for coordinating movement and balance, is proportionally very large relative to the overall brain volume, reflecting the extreme demands that flight places on motor coordination and the processing of vestibular, visual, and proprioceptive information that must be integrated in real time during aerial locomotion. The visual system of the chicken is similarly elaborate, with 4 types of cone photoreceptors providing tetrachromatic color vision that extends into the ultraviolet range, and with a specialized region of the retina called the fovea that provides high-acuity vision for detecting objects at close range during foraging. These neural features are not modern inventions in the chicken lineage, but inherited characteristics of the broader avian lineage, conserved across the entire bird radiation and traceable, through comparative analysis, to the visual and motor neural elaborations that characterize the small-brained but behaviorally complex theropods of the Cretaceous. The brain of a chicken is not a simple thing. It is the current expression of a neural design that has been refined across 165 million years of archosaur evolution, and its apparent simplicity, relative to a mammal brain, is in part an artifact of the very different evolutionary path that produced it. Simple is not always what it seems. The digestive system of a chicken preserves, in its basic organization and functional chemistry, the blueprint of a digestive system that was already present in the theropod ancestors of birds, modified in its details by the specific dietary adaptations of the avian lineage, but conservative in its fundamental architecture, in the way that all core physiological systems tend to be conservative, changing slowly and incrementally, rather than undergoing wholesale redesign with each evolutionary transition. The gizzard, the muscular stomach of birds that mechanically grinds food in the absence of teeth, is one of the most distinctive features of the avian digestive system, and one of the most clearly adapted structures in the entire chicken anatomy, capable of generating crushing forces sufficient to grind hard seeds, small stones, and even bone fragments into particles small enough for chemical digestion in the intestine. The gizzard is not a new invention, but a highly modified version of the muscular stomach present in all vertebrates, elaborated in birds in connection with the loss of teeth and the adoption of dietary strategies that require mechanical processing of hard food items before chemical digestion can proceed effectively. The loss of teeth in the bird lineage, which occurred gradually across the early evolution of birds with different tooth morphologies persisting in different lineages, until the early Cretaceous, was compensated by the elaboration of the gizzard, a trade of one grinding mechanism for another, and the chicken that swallows a pebble to help its gizzard work is following an ancient behavioral prescription written into its digestive biology long before the first true domestic chicken was ever kept. The body remembers what it gave up and makes do with what it kept. The circadian clock system of birds, the molecular machinery that generates and maintains the approximately 24-hour biological rhythms that govern sleep, feeding, hormone secretion, and many other physiological processes, is built from the same core set of clock genes that are found in virtually all multicellular animals, from fruit flies to humans. And its conservation across such an enormous evolutionary distance speaks to the fundamental importance of circadian timing for all complex life. In birds, the circadian system has some distinctive features compared to mammals, including the presence of light-sensitive cells, not just in the eyes, but in the pineal gland and in the brain itself, allowing some birds to entrain their circadian rhythms to light cycles even when their eyes are covered. A capacity that has no equivalent in mammals, and that may be related to the particular challenges of maintaining circadian coordination in migratory birds that experience rapidly changing light environments. The pineal gland of birds, which produces melatonin in a pattern that reflects the light-dark cycle, is a more directly light-sensitive organ than the mammalian pineal, and its prominent role in avian circadian regulation may reflect the ancestral condition of the archosaur pineal gland, retained in birds while being modified in the mammalian lineage toward the indirect, multisynaptic pathway that characterizes the mammalian circadian system. When a chicken settles onto its roost as the light fades and its melatonin levels begin to rise, it is responding to the same molecular signal, melatonin released by the same ancient glandular structure, that animals have used to track the rhythm of night and day since long before the first dinosaur ever walked the earth. The clock that ticks inside the chicken ticked inside the monster too. The feather coloration of the red junglefowl, the wild ancestor of the domestic chicken, is produced by a combination of pigment-based and structural coloration mechanisms that together create the iridescent, richly patterned plumage that serves multiple social functions in the wild bird, from mate attraction to species recognition to individual identification. And the study of feather pigmentation has provided another line of molecular evidence connecting modern birds to their dinosaurian ancestors. Melanin pigments, which produce the black, brown, and reddish-brown colors found in the plumage of the red junglefowl and many other birds, are synthesized in specialized cells called melanocytes that migrate to the developing feather follicle from their embryonic origin in the neural crest, a population of multipotent stem cells that also gives rise to many other cell types including neurons, cartilage cells, and glandular cells. The preservation of melanosomes, the cellular organelles that contain and store melanin, in fossil feathers from the Cretaceous and Eocene, has allowed paleontologists to reconstruct the colors of some extinct birds and feathered dinosaurs with considerable confidence, because the shape of melanosomes is correlated with the type of melanin they contain and hence with the color they produce. Sinosauropteryx, a small feathered dinosaur from the Chicxulub Formation, had chestnut and white banded coloration on its tail, reconstructed from the shapes of its preserved melanosomes, and this reconstruction demonstrates that the color-producing cellular machinery of modern birds was already present and functional in non-avian theropods more than 100 million years ago. Color is not just beauty, it is evidence. The insulin signaling pathway of chickens and other birds shows a distinctive modification compared to the equivalent pathway in mammals, a modification that has practical implications for understanding metabolic disease in modern humans, and that simultaneously illuminates the evolutionary divergence between the avian and mammalian lineages in their approaches to blood glucose regulation. Birds in general, and chickens in particular, have blood glucose levels that are substantially higher than those of mammals of equivalent body size, approximately twice the mammalian level. Yet they do not develop the kind of chronic complication associated with elevated blood glucose in humans, and understanding the molecular basis of this tolerance has become a subject of significant biomedical interest. The avian insulin receptor shows structural differences from the mammalian receptor that alter its downstream signaling properties, and the tissues of birds respond to insulin in ways that are calibrated to the different metabolic demands and glucose homeostasis requirements of an animal with fundamentally different energy metabolism than a mammal. These differences in glucose metabolism may be connected to the extreme energy demands of flight, which requires rapid mobilization of glucose from liver glycogen stores and efficient delivery of that glucose to the flight muscles, and the metabolic adjustments that support flight physiology in flying birds may have been retained in domestic chickens, even after those birds lost the need to fly. The chicken's unusual blood sugar is a metabolic signature of an aerial lifestyle, preserved in an animal that no longer takes to the air, a physiological memory of a capacity that domestication has suppressed but not erased. The body carries forward what the life no longer requires. The vocalization system of the domestic chicken, which produces a repertoire of dozens of distinct calls with specific social functions, ranging from alarm calls that distinguish aerial from ground predators to food calls that attract conspecifics to a resource, represents a level of acoustic communication complexity that was not fully appreciated until the detailed behavioral studies of the past several decades brought it into focus. The alarm call system of chickens is particularly remarkable because hens and roosters produce acoustically distinct calls in response to aerial predators like hawks and ground predators like foxes, and other chickens respond appropriately to each call type, taking cover from aerial threats and standing alert and scanning for ground threats, demonstrating that the call system encodes meaningful information about the nature and location of danger, rather than simply expressing an undifferentiated alarm state. This referential signaling, in which a vocalization points to a specific category of external event, rather than simply reflecting the caller's internal emotional state, is a feature shared with some primates and other animals considered cognitively sophisticated, and its presence in chickens suggests that the cognitive preconditions for complex communication were present in the theropod lineage long before anything resembling human language appeared on Earth. The rooster that calls out a warning when a shadow passes overhead is doing something that is, in its basic computational structure, not entirely unlike what a person does when they shout a warning to a friend. The gulf between them is real, but it is smaller than it looks from a comfortable distance. The social structure of domestic chicken flocks, organized around the dominance hierarchy, known colloquially as the pecking order, a term that actually originated from scientific studies of chicken social organization in the early 20th century, and was subsequently adopted as a metaphor for human social hierarchies, reflects an ancestral social organization system that was probably present in the wild jungle-fowl progenitors of domestic chickens, and that may have deep roots in the social behavior of the theropod lineage more broadly. Linear dominance hierarchies, in which each individual knows its rank relative to every other individual in the group and behaves accordingly, are found in many bird species and in many mammal species, and they represent a solution to the problem of organizing social groups in ways that reduce the costs of constant conflict by establishing predictable expectations about who defers to whom in any given encounter. The cognitive demands of maintaining a linear dominance hierarchy scale with the size of the group, because an animal must recognize and remember its relationship to every other individual it encounters, and the ability to maintain these social memories and use them to guide behavior in real time, requires a brain capable of sophisticated social computation. Chickens maintain recognizable dominance hierarchies in flocks of up to approximately 70 to 80 individuals, a social cognitive feat that goes largely unremarked precisely because it is so familiar, but which represents a level of individual recognition and relationship tracking that most people would be surprised to find in an animal they associate primarily with dinner time. The pecking order is not a simple thing. It is a society, remembered one relationship at a time. The sleep physiology of birds offers one final and particularly resonant line of evidence, connecting the biology of modern birds to the deep evolutionary heritage of the archosaur lineage. Because birds, like mammals and unlike other reptiles, experience a sleep state characterized by rapid eye movements and associated with the kind of high-frequency brain wave activity that in mammals is linked to dreaming. A finding that suggests that the neurological basis of REM sleep evolved before the divergence of birds and mammals from their common amniote ancestor, or that it evolved independently in both lineages, either of which has profound implications for our understanding of the function and evolutionary history of this mysterious state of consciousness. Birds experience sleep in a particularly interesting way because, as animals that must often remain vigilant against predators even while resting, many species have developed the ability to sleep with one eye open, literally, by allowing one hemisphere of the brain to enter sleep, while the other hemisphere remains awake and monitoring the environment, a state called unihemispheric slow-wave sleep that has no equivalent in mammals. This asymmetric sleep, in which half the brain rests while the other half watches, is connected to the asymmetric visual processing system of birds, in which each eye projects primarily to the opposite hemisphere, and it allows a bird on a roost to monitor its surroundings while still achieving the restorative benefits of sleep. A sleeping chicken on its perch, one eye perhaps half open in the dimness, is performing a neurological feat that humans can barely imagine, resting and watching simultaneously, the two states coexisting in a brain divided by the ancient demands of survival. Even in sleep, the old vigilance persists. The warmth of a sleeping bird on its roost, the particular quality of rest that belongs to an animal with a body temperature higher than your own, breathing with a system more efficient than yours, dreaming perhaps in ways you cannot fully imagine, is a warmth that carries within it the entire history we have been tracing tonight. A warmth that connects directly and without interruption to the metabolic fires of the Cretaceous, to the warm blood moving through the bones of animals that have been gone for 66 million years and yet are not entirely gone at all. Let that warmth move through you now, passing from the ancient world through all the long corridor of time into this quiet room where you are resting. Your breath is slow. Your body is heavy. The facts have been accumulating like sediment, layer upon layer, and now they are simply there beneath you, solid and still, and you are floating on the surface of all that accumulated knowledge with nothing required of you except to rest. The birds are asleep on their perches in the dark outside your window, and their dreaming is very old, and yours can be too. Let it be old. Let it be easy. Let the warmth of 66 million years of survival hold you where you are. Ancient DNA, the genetic material recovered from the remains of organisms that lived and died long before the invention of modern molecular biology, has transformed paleontology from a science of shapes and structures into a science that can, in favorable circumstances, read the actual genetic instructions that built extinct animals and governed their biology while they were alive. The field of ancient DNA research began in the 1980s with the recovery and sequencing of short fragments of genetic material from preserved museum specimens, and it has expanded with breathtaking speed as sequencing technology has improved, reaching the point where complete genomes can now be recovered from specimens tens of thousands of years old under the right preservation conditions. The oldest reliably authenticated ancient DNA sequences come from permafrost-preserved specimens in the Arctic and subarctic, where the combination of cold temperatures, low humidity, and absence of oxygen creates conditions that slow the chemical degradation of DNA to a rate that allows meaningful quantities of readable genetic material to survive across timescales of hundreds of thousands of years. The study of ancient DNA from extinct megafauna like woolly mammoths, cave lions, and Neanderthals has revealed population histories, migration patterns, and biological characteristics that could not have been inferred from bones and stones alone. And it has demonstrated with stunning clarity that the genetic information encoded in the molecules of life can survive the death of the individual organism by a margin of time that the human mind struggles to fully accommodate. The molecule that carries the instructions for life is more durable than life itself. The degradation of DNA in biological remains follows well-understood chemical pathways that set hard physical limits on the maximum age from which genetic material can be recovered in any form. And understanding these limits is essential for evaluating the claims that are sometimes made about the recovery of dinosaur DNA. Claims that the science, considered carefully, does not support. DNA degradation begins immediately after the death of an organism, driven by the action of enzymes released from disrupted cells, by microbial activity, by hydrolysis, which is the chemical reaction of DNA with water that breaks the bonds between nucleotides, and by oxidative damage from reactive oxygen species. Under optimal preservation conditions, specifically cold, dry, dark, and anoxic environments, the rate of hydrolytic depurination, which is the primary chemical pathway of DNA degradation, has been estimated to limit the survival of readable DNA sequences to a maximum of approximately 1 to 2 million years, and most researchers consider sequences older than several 100,000 years to be essentially unrecoverable in any biologically meaningful form. This limit, established by the chemistry of DNA itself, rather than by any limitation of our current technology, means that dinosaur DNA, from animals that died a minimum of 66 million years ago, is approximately 100 times too old to have survived in any readable form, regardless of the preservation conditions of the fossil. The chemistry is unambiguous on this point, and no amount of technological sophistication can recover information that the laws of chemistry have already destroyed. The genome of a dinosaur exists now only in its living descendants. The protein collagen, as we have already touched upon, is substantially more durable than DNA under geological preservation conditions, because its triple helix structure and its intimate association with the mineral hydroxyapatite of bone create a physical and chemical environment that protects it from degradation at a rate that exceeds the protection available to the more chemically labile DNA molecule. Studies of collagen degradation rates under various temperature and humidity conditions have suggested that collagen fragments may survive in fossil bone for tens of millions of years under favorable conditions, and the reported recovery of collagen peptides from Cretaceous dinosaur specimens, while still debated in some quarters of the scientific community, is not ruled out by the same thermodynamic arguments that make DNA recovery from such specimens impossible. The amino acid sequences preserved in dinosaur collagen, while far less information-rich than a full genome sequence, still carry evolutionary signal because the specific amino acids present at specific positions in the protein chain are determined by the genetic code of the organism that produced them, and comparing those sequences to the equivalent collagen sequences of living animals allows the evolutionary relationships of the extinct animal to be assessed. The finding that T. rex collagen sequences are most similar to those of modern birds, particularly chickens, is not a curiosity. It is a molecular phylogenetic result that independently confirms what the anatomy of the fossil has been telling us for decades. The protein and the bone tell the same story. They always have. The science of paleoproteomics, the recovery and analysis of ancient proteins from fossil material, represents the leading edge of molecular paleontology and extends the temporal reach of biomolecular analysis well beyond the limits imposed by DNA degradation, potentially into the range of tens of millions of years for specimens preserved under favorable conditions. The most significant advance in paleoproteomics in recent years has been the development of mass spectrometry techniques capable of identifying and sequencing protein fragments at levels of sensitivity and precision that were simply not achievable with earlier technology, allowing the detection of protein remnants that exist at concentrations far below what conventional biochemical methods could detect. A landmark study published in 2019 reported the recovery of protein sequences from a Homo antecessor specimen approximately 800,000 years old. At the time, the oldest protein sequences ever recovered from a hominin, and the phylogenetic analysis of those sequences provided evidence about the evolutionary relationships of this ancient human species that could not have been obtained from the morphology of the fossils alone. The same methodological approach, applied to dinosaur specimens, has produced the collagen results described earlier, and ongoing research is attempting to push the boundaries of paleoproteomics further into deep geological time to assess what, if anything, of the original protein complement of dinosaur tissues might still be recoverable after 66 million years. Each new methodological advance extends the reach of the living world into the past by another increment, and the past, it turns out, is not as silent as it once appeared. It has been waiting to be asked the right questions. The concept of dinosaur de-extinction, the idea of using genetic and developmental biology techniques to recreate living non-avian dinosaurs from the genetic and biological material available in their living bird descendants, has captured the popular imagination at least since the publication of Michael Crichton's novel in 1990 and the film adaptation that followed. But the scientific reality of what such a project would require is considerably more complex and considerably more. The fictional premise that dinosaur DNA could be recovered from blood-feeding insects preserved in amber and used to clone living dinosaurs, founders immediately on the chemistry of DNA degradation discussed above, because no insect preserved in amber 66 million years ago contains recoverable DNA from any source, dinosaur or otherwise, and no cloning technology can reconstruct a genome from a molecule that no longer exists in any readable form. But the fictional premise, while scientifically untenable, points toward a genuinely fascinating scientific question that researchers are actively pursuing through very different means. The question of whether the developmental genetic programs that built a dinosaur can be partially reconstructed and reactivated in the developing embryo of a living bird, producing an animal that expresses some of the ancestral characters of its dinosaurian lineage in its own body. The answer, it turns out, is a qualified and surprising yes. The genome of a chicken still contains the instructions for building a dinosaur. They have simply been turned off by 66 million years of evolution, and turning them back on, at least partially, is something that developmental biologists have already begun to do. The atavism, which is the reappearance in an individual organism of a trait characteristic of a distant ancestor that has been absent in the lineage for many generations, represents one of the clearest demonstrations that the developmental genetic programs of ancestral forms are not deleted from the genome when the traits they produce become evolutionarily suppressed, but are instead retained in a dormant form, regulated into silence by changes in gene expression patterns that can, under the right circumstances, be reversed. Atavisms have been documented in many species, including occasional horses born with extra toes reminiscent of their multi-toed ancestors, occasional whales born with hind limb-like appendages reminiscent of their terrestrial ancestors, and most relevant for our purposes, occasional chickens and other birds born with teeth. Yes, teeth. Several documented cases of chickens with tooth-like structures in their beaks have been recorded in the scientific literature. An experimental manipulation of signaling pathways in chicken embryos has been shown to reliably induce the formation of tooth-like structures, demonstrating that the genetic program for tooth development is not absent from the chicken genome, but is suppressed by the regulatory changes that accompanied the loss of teeth in the avian lineage. The sleeping instructions for building a dinosaurian tooth are present in the genome of every chicken alive, waiting in the silence of an unused regulatory state, and the right molecular signal can wake them. The chicken carries its ancestry not as history but as potential. The laboratory of Jack Horner at Montana State University, and subsequently at Chapman University, working in collaboration with developmental biologist Hans Larson at McGill University, has pursued the most sustained and publicly prominent program of research aimed at understanding the developmental genetics of dinosaurian characters in living birds, with the explicit long-term goal of producing a chicken embryo that expresses a suite of ancestral dinosaurian traits. An animal that its creators informally call a Chicxulub, or a dino chicken. The research program is not science fiction. It is a serious scientific enterprise grounded in the rapidly advancing understanding of the genetic and molecular controls of vertebrate development. And it has already produced concrete results in the form of chicken embryos with modified developmental programs that express more dinosaur-like anatomical features than the unmodified wild type. Manipulating the expression of specific genes during the development of chicken embryos has produced embryos with elongated dinosaur-like snouts instead of shortened avian beaks, with more ancestral wrist structures similar to those of non-avian theropods, and with tail developmental programs that more closely resemble the long, bony tails of dinosaurs than the short, fused pygostyle of modern birds. These experiments are not creating dinosaurs. They are revealing the developmental architecture that connects the living chicken to its extinct relatives, demonstrating with molecular precision that the distance between a chicken and a dinosaur is, in some senses, smaller than the distance between a chicken and what we casually assume a chicken to be. The chicken contains the dinosaur. The dinosaur is in the developmental code. The beak of a bird is one of the most distinctive and defining features of the entire class, and understanding how the beak evolved from the toothed jaws of theropod dinosaurs has been a major focus of evolutionary developmental biology research in recent years, producing results that are as surprising as they are illuminating. The beak is not simply a toothless jaw. It is a fundamentally restructured facial skeleton in which the premaxilla, the bone that bears the front teeth in most vertebrates, has expanded to form the bulk of the upper beak, while other facial bones have been reduced or fused in a pattern that has no exact equivalent in any non-avian dinosaur. Research by Bartanjan Buller and Arhat Abjanov and their colleagues has traced the developmental genetic basis of this restructuring to changes in the expression of specific signaling molecules during facial development. And they demonstrated in a remarkable experiment that reverting the expression of these molecules to a more ancestral pattern in chicken embryos produces embryo faces with more dinosaur-like snout morphology, longer and narrower rather than short and wide. Similar to the facial proportions of small-snouted dinosaurs like Velociraptor, the experiment did not produce a velociraptor. It produced a chicken embryo with an unusual face and a great deal of scientific information about the developmental basis of one of the most important evolutionary transitions in the history of vertebrate life. Every beak is a message about where it came from. Now we are learning to read the message backwards. The tail of a bird, reduced in the course of avian evolution from the long bony tail of theropod dinosaurs to the short fused structure called the pygostyle that supports the fan of tail feathers in modern birds, represents one of the most dramatic skeletal transformations in the entire bird-dinosaur transition. And the developmental genetic basis of this transformation has been studied in chicken embryos with results that reveal the remarkable persistence of ancestral developmental programs in the living genome. In the early stages of chicken embryonic development, the tail grows as a long, segmented structure with a morphology more reminiscent of a dinosaur tail than of the final adult form, with clearly defined individual vertebrae extending well beyond the body. This elongated embryonic tail is subsequently shortened and fused during later development, as the caudal vertebrae are reduced in number, and the terminal vertebrae fuse to form the pygostyle. And this transformation is under the control of specific molecular signals, whose disruption produces chickens with more elongated, dinosaur-like tails. The fact that the embryonic chicken grows a dinosaur tail and then suppresses it during development is one of the most vivid demonstrations available of the principle that evolutionary change often works not by eliminating ancestral developmental programs, but by modifying their timing, duration, and outcome, leaving the ancestral program present and partially active in the genome, even after the adult morphology it originally produced has been replaced by something new. The dinosaur tail grows inside every chicken egg. It is just that development has learned to take it back before hatching. The microbiome of modern birds, the community of bacteria, archaea, fungi, and viruses that inhabit the gut and other tissues of living birds, represents a largely unexplored dimension of avian biology, with potential implications for understanding the evolution of bird physiology, disease ecology, and the complex relationships between vertebrate hosts and their microbial passengers across deep evolutionary time. The gut microbiome of chickens has been studied in some detail because of its practical importance for poultry health and production. And what these studies have revealed is a complex and dynamic community of microorganisms that varies with diet, age, health status, and environment, and that plays important roles in nutrient extraction, immune system development, and resistance to pathogens. The ancestral microbiome of the dinosaurian lineage is, of course, entirely inaccessible to direct study, because microbial cells leave no reliable fossil record, but comparative studies of the gut microbiomes of birds and their closest living relatives, the crocodilians, have identified some shared features that may represent conserved elements of the ancestral archosaur microbiome, transmitted through the lineage across hundreds of millions of years alongside the host genome. The microbiome is not separate from the evolutionary story of the bird. It is part of that story, a community of partners whose own evolutionary history is intertwined with the history of the animal that carries them. And the bacteria fermenting plant material in the gut of a chicken today may be the distant descendants of bacteria that performed the same service in the guts of their dinosaurian hosts. Even the invisible passengers have been making the journey all along. The question of dinosaur cognition, of what it was actually like to be a non-avian dinosaur from the inside, is one that paleontology cannot answer directly, but that cognitive science and comparative neuroscience can approach obliquely. By studying the brains and behaviors of the living animals most closely related to non-avian dinosaurs, and using that information to make educated inferences about the cognitive capacities of their extinct relatives. Birds, and particularly the most cognitively sophisticated birds, the corvids, the parrots, and the raptors, demonstrate problem-solving abilities, causal reasoning, tool use, planning for future needs, and in some species a capacity for recognizing themselves in mirrors that is otherwise found only in great apes, dolphins, and elephants among non-human animals. These cognitive abilities are not products of mammalian brain architecture, but of a fundamentally different neural organization, one in which the functions performed by the layered cortex of the mammalian brain are accomplished in birds by a densely packed nuclear structure called the pallium, organized differently but capable of equivalent computational feats. The implication for our understanding of non-avian dinosaurs is that the small-brained but cognitively flexible theropods that gave rise to birds may have been capable of more sophisticated behavior than their relatively small brain volumes alone would suggest, if those volumes were organized in a similarly efficient nuclear pattern. We do not know what it was like to be a dinosaur, but we can be fairly confident that it was not the dim, automatic, stimulus-response experience that the old reptilian model implied. Something was happening in those ancient heads. Something was watching the world and making sense of it. The field of synthetic biology, which involves the design and construction of new biological parts, devices, and systems, as well as the redesign of existing natural biological systems for useful purposes, has begun to intersect with paleontology in ways that promise to blur the boundary between studying the past and recreating aspects of it in living biological systems. Researchers have used synthetic biology approaches to reconstruct ancestral protein sequences from the inferred evolutionary ancestors of modern proteins, by using phylogenetic analysis to work backward from the sequences of living proteins to the sequences of their common ancestors, and then synthesizing DNA, encoding those ancestral sequences, and expressing the ancestral proteins in living cells to study their properties. This approach, called ancestral sequence reconstruction, has been used to recreate proteins from organisms that lived hundreds of millions of years ago, including ancestral elongation factors from the common ancestor of all life, ancestral fluorescent proteins from the ancestors of modern corals, and ancestral steroid hormone receptors from the ancestors of modern vertebrates. Applying this approach to proteins whose modern forms are found in both birds and other vertebrates allows the properties of those proteins at different points in evolutionary history to be inferred and tested, providing a window into the molecular biology of extinct animals that complements the information available from fossil morphology and preserved ancient proteins. The past can be rebuilt, molecule by molecule, in a laboratory, and what is rebuilt is not a ghost but a functional piece of biochemistry that once operated inside a living animal. Science is learning to run evolution in reverse, one protein at a time. The possibility of using CRISPR-Cas9 gene editing technology to introduce ancestral genetic variants into the genome of a living bird, thereby recreating in a living animal some of the genetic conditions that characterized an ancestral form, is one that developmental biologists and evolutionary geneticists have discussed with increasing seriousness as the technology has matured and the relevant comparative genomic data has accumulated. CRISPR-Cas9 allows specific DNA sequences to be targeted and modified with unprecedented precision, and the identification through comparative genomics of specific genetic changes that distinguish the genome of a chicken from the inferred genome of its non-avian dinosaur ancestor provides a list of candidate modifications that could, in principle, be introduced into a chicken embryo to test their developmental effects. This is not the same as resurrecting a dinosaur. It is something more subtle and in many ways more scientifically interesting: the systematic exploration of the genetic basis of evolutionary change by observing what happens when specific changes are reversed or modified in a living developmental system. The results of such experiments, were they to be carried out comprehensively, would not produce a velociraptor or a T-Rex. They would produce a spectrum of modified chickens, each one illuminating a specific aspect of the genetic architecture of the bird-dinosaur transition, and each one demonstrating in living flesh and blood the principle that the distance between what a chicken is and what a dinosaur was is measured not in an unbridgeable gulf of biological impossibility, but in a specific set of genetic changes that are, at least in principle, known, countable, and reversible. The chicken is a dinosaur with its options constrained. Evolution constrains options. Science opens them again. The ethical dimensions of de-extinction science, whether applied to relatively recently extinct species like the passenger pigeon or woolly mammoth, or to the more speculative project of recreating dinosaurian characters in living birds, are as complex and as genuinely unresolved as the scientific challenges themselves, and they deserve to be considered with the same rigor and intellectual seriousness that the scientific questions receive. The creation of an animal that expresses ancestral dinosaurian characters in a body derived from a domestic chicken raises questions about the welfare of the animal itself, about the ecological consequences of introducing such an animal into a world that has not contained its like for 66 million years, and about the appropriate limits of human intervention in the processes of life and evolution. These are not questions that science alone can answer, because they involve values as well as facts, and the values that different people bring to them are genuinely different in ways that cannot be resolved by the accumulation of additional data. What science can do, and what it has done with increasing power and precision over the past century, is to clarify what is actually possible, to replace the vague and often inaccurate intuitions that people bring to these questions with precise knowledge of what the relevant technologies can and cannot achieve, and to ensure that the ethical deliberation, whatever form it takes, is grounded in an accurate understanding of the biological reality it is deliberating about. Whether we should try to make a dino chicken is a question for society. Whether we can is a question that is already being answered, and the answer is more yes than most people realize. The paleontological legacy of the bird-dinosaur connection extends beyond the purely scientific into the cultural and philosophical, because the recognition that birds are living dinosaurs fundamentally changes the relationship between the human species and the animals we share our world with, revealing a depth of evolutionary time and a complexity of biological heritage that the ordinary human encounter with a bird at a feeder or a chicken at a farmyard does not obviously suggest. To look at a bird with the knowledge that it is a dinosaur is to look at it differently, to see in the lightness of its bones the hollow pneumatization of its Cretaceous ancestors, to see in the efficiency of its breathing the air sac system that once sustained animals that weighed 10 tons, to see in the organization of its genome the compressed and rewritten library of 165 million years of theropod evolution. This kind of knowledge does not diminish the beauty of a bird. It increases it by adding to the visual beauty of feathers and form the intellectual beauty of deep time and evolutionary continuity. The sparrow on the branch outside your window is not just a sparrow. It is the current edition of an animal whose ancestors survived the most destructive single event in the past 66 million years, whose lineage has been continuously present on Earth since before the first flower bloomed, and whose genome contains, in its regulatory sequences and its conserved coding regions, a compressed record of virtually every major event in the evolutionary history of vertebrate life. A sparrow is the universe looking at itself through the eyes of a creature that survived everything. That is not a small thing to be. The future of our understanding of the dinosaur-to-bird transition is brighter than it has ever been, because the tools available to investigate that transition, from high-resolution CT scanning of fossil specimens to single-cell genomics of developing bird embryos to the ancestral sequence reconstruction of ancient proteins, are improving at a rate that makes predictions about what will be known in 10 or 20 years essentially impossible. New fossil sites are being discovered and excavated continuously, particularly in China and in other parts of Asia and South America, where the Mesozoic geological record is rich and relatively unexplored. And each new discovery has the potential to fill a gap in the evolutionary sequence, or to reveal an unexpected twist in the story that forces a reconsideration of assumptions that seemed firmly established. The genomic sequences of more and more bird species are being determined and compared, building a comparative database that allows the identification of conserved regulatory sequences, ancestral gene expression patterns, and the molecular signatures of evolutionary events that occurred tens of millions of years ago in the ancestors of modern birds. The developmental biology of bird embryos is being studied at a level of molecular detail that was simply not achievable even a decade ago, revealing the genetic architecture of the transition from dinosaurian to avian anatomy, one regulatory interaction at a time. The science is alive and moving, and the story it is telling is not finished. Every year the picture becomes clearer, the connections more precise, the ancient world more legible. We are learning to read the deep past in the bodies of the living present, and what we are learning is that the present contains the past more completely and more faithfully than anyone who studied these questions a century ago could have imagined. The dinosaurs are not gone. They are singing outside your window, and their song is 66 million years old. The last fact in our journey tonight is the simplest, and in some ways the most astonishing of all, a fact that requires no technical vocabulary, no genomic sequencing, no isotope chemistry, no electron microscopy, no developmental manipulation of embryonic signaling pathways, nothing more than a willingness to sit with an ordinary observation and follow its implications wherever they lead. The fact is this: a chicken is alive. It breathes, it regulates its body temperature, it perceives the world through senses more refined than our own in some dimensions. It communicates with others of its kind using a vocabulary of dozens of meaningful signals. It dreams, if our best evidence is to be believed. and it carries within its cells a genome that connects it without interruption to animals that walked the earth when the continents were in different positions, and the atmosphere had a different chemical composition, and nothing that would one day become a human being had yet appeared anywhere on Earth. A chicken is alive, and its life is not separate from the history of life, but is continuous with it, an unbroken thread stretching from the first self-replicating molecules in some ancient warm pool, all the way to the bird that is somewhere right now tucking its head beneath its wing in the dark. That continuity is the most astonishing thing about life, more astonishing than any individual fact or mechanism or discovery. The simple astonishing fact that it has kept going, that it has found a way through every catastrophe and every extinction and every impossible challenge that deep time has placed before it, and that it is still here, still going, still making new things out of old instructions, still finding ways to be alive in a universe that does not particularly care whether it succeeds. Life succeeded anyway. The chicken is the proof. The chicken was always the proof. And now the last of the ancient world settles gently around you, like the finest sediment finding its final resting place after a long journey through dark water. And there is nothing left to carry forward into this night, except the quiet weight of knowing that the living world is deeper and stranger and more continuous than the surface of things suggests. The birds outside are silent now, or nearly so, their ancient dreams moving through minds that have been dreaming since before our kind existed. And the darkness is the same darkness that fell over the first forests after the last great light. The darkness in which small feathered survivors waited out the long cold and found, eventually, that morning had returned. Your breath is the breath of an animal that also survived, also adapted, also carried its ancient chemistry forward through impossible time to this particular moment, this particular room, this particular quiet. The story of life is the story of persistence, told in the language of bone and feather and gene and breath. And tonight you have heard a portion of it, and now you may rest inside it, cradled by the long continuity of everything that made you possible. This has been learned when you sleep. Good night.